When I was doing PhD, he was doing MSc at that time. Then Dr. Indu is there. He was my student. Dr. Vikas is there. Dr. Vikrant is there. So I am very happy to see them all rising. So coming to the talk, that is the climate resilience. How science and technology is responsible for this resilience. Can I have some light here? Anyhow, no. Thank you. Now coming to the topic, in the recent time, the human activities are fast disturbing the natural ecosystem. As I have just told you, that our climate is bearing all the disturbances. And the main cause for these disturbances is the human activities. Whatever the activities are there, whatever the fertilizer we are using, whatever the fossil fuels we are consuming, or whatever the pesticides, fungicides we are spraying on the crops, all these are responsible for this climate change. So the climate change, they are putting stress on the agricultural crops. Every crop, has to be sprayed with the pesticide, fungicides, insecticides. The important climate-based abiotic stresses in the plants are like drought or water scarcity. Now, due to this climate change, all these crops, they are facing the water scarcity, salinity, then excessive heat in the summer and cold in the winter. Now you can see nowadays excessive cold is there. In the summer season, you will see that excessive hot season is there. Unbearable temperature in the summer season is there. And so the crops, they are also unable to bear all these climatic changes. It is a challenge to science and technology to develop climate resilient crops which can fight drought, heat, cold, and the soil toxin. Now, the plants or the crops, they are living being like us. As we are breathing, they are also breathing. As we are uh, doing the metabolic activities, they are all doing the metabolic activity, and they need the congenial environment. But due to the human activities, this congenial environment that is not available to our crops. That is why the scientists, they are searching and doing regular researches so that to improve the crops in order to face all these adverse environmental conditions. So the theme of this conference, as I have already told you, has nicely been selected by the organizing secretaries or organizers, bringing science and technology for climate resilience. So all these three topics, science, technology, and climate, as I have already told you, they are dependent on one another. So it is definitely going to benefit the finding out the ways and means to develop the climate adverse climatic resilient crops. The collaboration of biological sciences and technology gave rise to new field of science, biotechnology, which work in this direction. The climatic changes, they are converting our fertile agriculture into non-fertile by increasing the alkalinity, acidity, desertification, Then the biological or microorganisms, they play a very important role in the production of our crops, biological microorganisms. So they are being killed by the acidity of the soil, by the alkalinity of the soil, or when we put fertilizers, or when we put these pesticides, insecticides on the crop. So blue-green algae have the potentiality to reduce the acidity and alkalinity and reclamation of the agricultural land to restoring its fertility. So this blue-green algae, it is also the microorganism which is responsible to face the adverse climatic condition in order to increase the production of the crop plants. The agricultural scientists, they are highly concerned regarding climatic changes and making all out efforts to develop resistant crop varieties so as to feed the ever increasing population. When these crops they have to face the adverse climatic condition, naturally whole of their metabolic system is disturbed. And when the metabolic system of the plant body is concerned, then they are unable to produce more and more. Whereas our population is increasing day by day, and on the other hand, due to changes in the climatic conditions, the crop production is being reduced. So currently, our country is facing a challenge of producing the adequate food to meet the demands of ever increasing population from shrinking natural resources, intensification of agriculture through enhanced productivity and resource use efficiency has to be the main focus as competition for land and water are increasing from non-farming sectors. Further, in recent years, the climate change and its vulnerability are emerging a major challenges to Indian agriculture. In view of these, immediate thrust is needed on enhanced production with reduced natural resources under a variable climate. So, this climate resilience, that is the very, very important burning topic of these days. The development and identification of climatic resistance crop varieties with enhanced tolerance to heat, drought, flooding, chilling, salinity stresses, they are very, very essential in order to sustain the improved crop yields to cope up with the challenges of the climatic changes. So it means heat. We have to reduce or we have to reduce the global warming, that is this heating effect on the plants. Then drought, we have to produce the drought resistant plants. Then flooding, flood resistant plants. Last year there were great floods in the Punjab and whole of the rice crop was destroyed. So we have to face all these situations. As a scientist, I can say that most of the scientists they are working in these type of the searches so as the plants they become resistant to the climatic changes, that is the climatic resilience. Climatic resilience, as you know, it is the ability to anticipate, prepare for and respond to hazardous events or disturbances related to the climate. So, once again, I will say that all these disturbances they are due to the human activity. We are responsible. All of us are responsible for this climatic change. Now, improving climatic resilience involves the assessing how climate change will create new or alters the current climatic related risk and taking steps to better cope up with these risks. So, how to protect our crops from all these risks of salinity, drought, acidity, etc. Then, climate resilient crops. They are the crops and the crop varieties that have enhanced tolerance to biotic and abiotic stresses. I think you are familiar with both these terms, biotic and abiotic stresses. In order to create mass awareness for adoption of climatic resilient technology, the Prime Minister of India on 28 September 2021 dedicated 35 crop varieties with special traits via video conferencing. The event was organized at all at the Indian Agricultural Research Institute, New Delhi, sorry, ICAR, Indian Council of Agriculture Research. These 35 new crop varieties with special traits such as climatic resilience and nutritional rich contents have been developed by ICAR to address the twin challenges of climatic change and malnutrition. These include a drought tolerance variety of chickpea, wilt, and sterility mosaic resistance pigeon pea, early maturing varieties of soybean, disease resistant varieties of rice, and biofortified varieties of wheat, pearl millet, maize, and chickpea, quinoa, buckwheat, winged bean, and faba bean. These special trait crops varieties, they also include those that address the anti-nutritional factor found in some crops that adversely affect the human and animal health. So in the last, I will say that it is not only the plants which are affected by this climatic change. It is the human being also and all the living organisms, even the smallest living organism, that is the viruses, they are also affected by this climatic change. So our duty is that whatever we learn from this conference, we should apply in our daily life. There are many activities which we can avoid or which we can perform in order to save our environment. So, with this brief introduction, I once again thank the Vice Chancellor and the organizers of this conference who have invited me to this conference. Thank you very much. Thank you.
Thank you so much, sir. Being a plant biochemist, I can feel the pain that is being faced by plants. And of course, our duty to take appropriate efforts to bridge science and technology for empowering sustainability. Thank you so much.
we have been graced by the presence of our chief guest of the day, Dr. Kanwar Deep Singh. He is HOD Virology Department, Medical College, Amritsar, and he is also Director of PIMS, Jalandhar. We welcome you, sir. Thank you for being a part of this conference. And I also would like to now, I think we should the next session. And through the virtual stage is available with us our next expert, Dr. O.P. Agrawal, who is the former Vice Chancellor, Jivaji University, Gwalior. He is specialized in the field of entomology, discipline of zoology. He has been awarded with Dad Fellowship, Germany, and many more awards. He supervised 27 PhDs and 26 MPhil students. He has more than 95 research publications in national and international renowned journals. Besides, he has vast experience of teaching and research. I invite Dr. O.P. Agrawal for his section. Over to you.
ताजगी भरा और इको फ्रेंडली नमस्कार. So I am presenting my lecture for this conference. The title of my lecture is Spirulina Superfood, Promise for Better Life and Better Environment. हिंदी में बोले तो स्पिरुलिना उच्च भोजन, बेहतर जीवन और पर्यावरण का वादा. So, may I have next slide, please? So this slide shows the figure of Hippocrates, who is known as father of medicine. He said, "Let food be thy medicine, and medicine be thy food." अपने भोजन को दवा बना लो और अपनी दवा को भोजन बना लो. Spirulina superfood is a kind of algae grown in water. Now it is grown in artificial ponds, shallow ponds, as a commercial venture. So this spirulina superfood perfectly fits in this coat of Father Hippocrates. Next slide, please. So now we come to the point, what is spirulina? Wait a minute. Spirulina is a cyanobacteria. It is a kind of algae. So it is a blue-green algae. It is simple unicellular form of algae that thrives in warm, alkaline, and freshwater bodies. Its name is spirulina because when we look this under microscope, it is spiral in shape. spring-like structure or helix-like structure. That's why its name is spirulina. Now we come to history of spirulina. Most of the Indian people do not know about spirulina, what is spirulina, and why should they use or how they can use it as a food. So a German algal scientist, Dr. Darwin, discovered its existence and named it as spirulina. But it become well known to modern society, human society, civilization only during 1962 when Dr. Clement of France, he was visiting Africa for his studies. What he saw that the people living around Lake Chad of Africa, they are healthier than the other people of their community living away from the lake. And he saw that the people, they were harvesting, they were removing scum, or that is algal scum from the lake water, and they were eating it along with their staple food. He came back to his country and tried to identify it, and it was already identified, it was spirulina. So it was known for thousands of years, and tribal people were using it as food, but modern society came to know about spirulina only after the rediscovery of Dr. Clement. So extensive global studies have started from this point on various aspects of spirulina. In 1966, the French scientists developed method of its culture in artificial ponds and using a culture medium. And the name of scientist was Jaruk. And so still this Jaruk medium is very popular for cultivation of spirulina. In 1974, it is misprinted in this slide. WHO and FAO declared spirulina to be the best food for future. NASA called it a superfood. Next slide, please.
नेक्स्ट स्लाइड शायद एक और हाँ ये फिर से बोल नीचे के
This slide shows the value of spirulina. Nutritional value of spirulina. A comparison is given. Spirulina has got a maximum amount of protein, that is around 65% protein is present in spirulina. In peanuts, groundnuts, the protein percentage is about 25%. In cheese, so, it is about about 25% of protein is present in cheese. In milk, it is about 4%. In chicken, 22%. In soya, is around 15%. But sometimes whole soya, it is more than that. But still, it is higher in spirulina. In eggs, it is about 13%. So, next slide, please. Spirulina is a simple and primitive plant. It is unicellular blue-green algae It is also known as cyanobacteria. It is considered as algae. Normally, it is known as pond scum. That is the kai. In Hindi, we call it kai.
Spelling करके
So, kai word looks very ugly, and but the cells of these
ये सब कैसे
spirulina, they are joined together,
जादू
and
ये मैंने
these fibers they are spring-like.
स्क्रीन बंद हो गई
There are two names are there: Spirulina platensis and Arthrospira. There are two names: most ancient and promising food for animals and humans. So in 1981, US FDA approved production and marketing of spirulina as food supplement. And
मेरी उम्मीद चल रही है
as food and food supplement in USA, Europe, Japan, UK, India, Australia, China, New Zealand, and other countries was given. It is the best solution for food crisis. It ensures food security. It is perfect medicinal fuel and ideal example of organic farming. So it is excellent tool for malnutrition. Next slide, please. This is the slide showing the pond, well-developed, well-equipped pond for culture of spirulina, and these ponds are raceway ponds for commercial cultivation of spirulina. Next slide, please. Now, our question is why we should use spirulina? Because it is declared as best food for future, that is superfood. It is also known as miraculous plant, miraculous algae, magic food, aquatic miracle. In Hindi, we can call Jali Sanjeevani. It is best natural food available on earth. It has got more than 65 nutrients, including protein, all amino acids, essential as well as non-essential amino acids are present in spirulina in good amount. And these amino acids and protein are highly digestible. Up to 90% digestibility has been recorded for spirulina because spirulina is made up of unicellular cells and its cell wall does not contain complex polysaccharides. Polysaccharides are not present in cell wall. So it is easily digestible and assimilable. It has got sufficient amounts of vitamins, minerals, salts, mineral salts, omega-6 and 3 like fatty acids. It has got antioxidant, immune boosting, boosters, and phytonutrients. It is different and unique source of nutrients. It can be used as food supplement to fulfill our food deficiencies. We eat food almost 3 times a day, but our deficiencies are not fulfilled, and most of the people, take synthetic food supplements, vitamins, and minerals, and still their problems are not solved. So, daily use of spirulina as food supplement is good for health. It prevents number of diseases, delaying aging process, and it helps in treatment of diseases. It is not a medicine. It can be called a medicinal food because it helps us recovery from the disease. Moreover, it is alkaline food. It promotes health status of the body. Spirulina has prebiotic and probiotic activities, properties. And we all know now the prebiotic and probiotic drugs, they are essential part of the treatment given by our doctors. Spirulina cannot be compared with, अरे बोलो नहीं, Spirulina cannot be compared with any other established food because it is better than best. It is not only the best but better than best. सर्वोत्तम से उत्तम क्या? Spirulina. This name, this title we have given to spirulina. It is acidic food. It is alkaline food. Our body, to maintain good health of our body, we need alkaline food. But mostly we are eating acidic food. We must eat 80% of alkaline food and 20% of acidic food. But reverse is happening. We are eating 80% of acidic food and 20% of alkaline food. So we have to increase alkaline food, and spirulina is a alkaline food. This is given in this figure. Next slide, please. Now this slide shows that spirulina is not only alkaline food, but it is a source of functional food ingredients. What are the functional food ingredients? The functional food ingredients are the ingredients or biochemical parameters obtained from the food which help to maintain normal functioning of our body. So it is not like other food, it is more than that. It is a source of bioactive substances which can regulate normal body functions and can help to ward off their deficiency, help in prevention and cure of diseases. Spirulina is known as super antioxidant due to presence of myriad of these such factors. It boosts immunity also. Spirulina can be used as a source of functional food ingredients to be used against specific diseases. Example of such bioactive substances are beta-carotene is present in spirulina, astaxanthin, phycocyanin, phycobilins, and some more. Some parameters are present which act as bioactive substances which can open a new path for new drug discovery, particularly against cancer. Next slide, please. So, spirulina may be used as a prevention tool against a variety of cancers. Food spirulina can serve as food supplement to improve general health for prevention of diseases. It also helps in healing process by using conventional drugs. Spirulina reduces side effects of chemotherapy and radiotherapy. And during last World War in Hiroshima and Nagasaki, the people are still suffering from that effects. And in their country, in these places, the spirulina was allowed to reduce the radiation effects because it helps in removing the side effects of radiations as well as chemotherapy. So isolation and purification and testing of spirulina components like phycocyanin, phycotin, polysaccharide, new natural and safe drug discovery can be made. Spirulina is approved as food supplement. It's used for healing purposes, does not require permission from any authority, ethical organization, or even it does not require prescription of doctor because spirulina is a food. It is a superfood, concentrated food. So no permission of doctor is required. One can use spirulina as superfood for better health. More pinpointed studies should be conducted to authenticate its benefits. Next slide, please. So now the point comes, can spirulina ward off malnutrition from? My answer is yes. WHO also, according to WHO, child deaths below 5 years are largely due to malnutrition. Lot of work has been done in India and abroad on children of all age groups, and it was found it reduces the symptoms of malnutrition. I will cut short because I have to cover so many things, and each aspect needs a separate lecture. So malnutrition, the spirulina is the most, next slide, spirulina is the most powerful weapon which you can use use to change world. You can make a change in the world, and we need a world without malnutrition. So next slide, please. These are some पेपर क्लिपिंग्स। स्प्रुलिना बचाएगा देश को, बच्चों को, देश के बच्चों को कुपोषण से। There are many people and many organizations in India too. They are working on spirulina and against malnutrition. And this is a guy from Bengaluru. He is growing spirulina in his own farm, and he is using spirulina against child malnutrition. Next slide, please. This is an international foundation, the Antenna Foundation. Its branch is in India also. They also work on spirulina against malnutrition. Next slide, please. This is another institute, that is Sole Institute. They are also working on spirulina against malnutrition, and the spirulina can be consumed as a tablets, as a powder mixed with food ingredients, food stuffs, and capsules are also there. Then besides, other formulations and value-added products have been tried. They are commercially available, and many NGOs are using them. Spirulina against, so next slide, please. So this slide, the people in India who have done tremendous work on spirulina against malnutrition. This Professor Seshadri, Fox Ripley, and Dr. Hendrickson, they are the great, they have done great work on spirulina against malnutrition. In India, they have conducted a very good survey and experiment that if they provide spirulina to the children, 1 gram spirulina per day can bring significant change in child malnutrition within 2 weeks. And this lower figure is Dr. Pushpa Srivastav of Rajasthan University, Jaipur. She has done commendable work on spirulina. She, with the help of DST and some other agencies, she has established her own local culture in a village about 30 kilometers away from the Jaipur, and focusing on employing the SC/ST women in her project. And she taught them how to culture spirulina and how to use spirulina in farm nutrition. Next. So I salute Dr. Pushpa Srivastav for her commendable work. At present, she is alive and she is very old. We invited her at Jivaji University, Gwalior, and she has given very beautiful lecture on her work. Then next slide, please. This is again in Tamil Nadu. Chennai is the headquarter. Offer Spirulina is an NGO. They are also growing spirulina, making spirulina value-added food products. They are organizing camps, particularly in tribal and poor areas, slum areas, and they are distributing spirulina food to them free of cost, and they also organize exhibitions where they offer people to consume spirulina tea, spirulina drinks, many things, and then they try to sell it. So they raise funds and they help poor people against malnutrition. Next slide, please. Why people don't know about and use spirulina? Because it was not described our ancient literature. In Ayurveda, spirulina is not mentioned. Furthermore, even to the Western world, spirulina was not known to the civilized people until 1960. Around 1980, the lot of work was now available, and around 1980, the spirulina products are flooded in the market. Many companies have started spirulina culture. They are producing tablets, powder, then capsules. They are also manufacturing other forms. Some companies are selling frozen spirulina. But people are prejudiced. Still, when they come to know that it is a kai, it is a pound scum, they don't want to use it. Spirulina is color is not very good. It is not pleasant. Its smell and taste are also not so good. So they are not favoring trying to use it. Then how to use spirulina? Next. Similarly, many people do not use honey, because when they come to know that honey is a vomit product of vomit of honeybees, they don't want to use it. But it is also yet another superfood. So how to use spirulina? Spirulina flakes, spirulina crunchy are also available. So this slide shows what is our contribution. Around 50 years back, I got interested in spirulina. I have done lot of survey work on spirulina, and I have collected literature, I have collected scientific research papers. So I have tried to use information technology tool to convert spirulina commercial product or final product. So we try to develop spirulina value-added food products biscuits, protein powders, tomato soup powders. Even we have mixed spirulina in some spices, chaat masala, raita masala, pani puri masala. So we have mixed. So we use food technology techniques and we have blended spirulina in these foods so that their bad smell is not there. So we were trying to mask the taste, color, aroma, flavor with another food ingredients. Particularly, spinach goes well with sweet stuffs like sugar, jaggery, and honey. So, in future, it may prove to be very healthy, new and healthy food revolution. Next slide, please. So, I call it translational or applicational research. So, next slide, please. Technology transfer. So I have developed with the help of several people and students in the lab at home and at home also with family members. We have tried to make. अभी बाद में बात करता हूँ. So this slide shows the what is there in spirulina. So we have transferred our technology. We have transferred our formulae to young entrepreneur. So he tried to market them. He made a company, Acme Foods and Nutraceuticals, and he was selling green gold spirulina products. Next slide, please. This green gold spirulina foods, there were biscuits, capsules, 4 types of spices were there, 2 types of green gold spirulina plus powders were there, and tomato soup powder were there, and some more things were there. He tried to sell his products in supermarket also. The tablets, people are fond of tablets. They just take in the mouth and chew it pleasantly. So these figures are there. Next slide, please. Next slide. Yeah, this is also there. Then we try to popularize it. Spirulina as a supreme ingredient for cookery and bakery. So it can be several ways and means are there to use spirulina. Spirulina can be used as a capsules or tablets or powder mixed in other things. Spirulina crunchy. So several ways and means were used. Next slide, please. Then later on, that this company who started selling spirulina, it was merged and taken up by a company that is known as Sumayu Global. It is Banaras-based company, and they are selling some spirulina products. So I personally recommend either you should use spirulina powder, you can add on, mix in with food food. You can cook with a number of recipes I am suggesting to be used. But if you are not ready to use spirulina powder due to some constraints, you can use spirulina-based protein powders which are available in the market, and you can use them to blend with your food. Next slide, please. So these various recipes I have given with the help of ladies, with the help of family members, with the help of friends, with the help of Institute of Hotel Management in Gwalior. I collaborated with them and we have organized recipe contest over in their college for students. This slide shows that we organized recipe contest. Director of the institute is also there. So in this way, we have to popularize consumption of the spirulina sweet. It goes very well. Copra Barfi with the spirulina. Next, go on. And various such commercial formulations are available in international market in many countries. Spirulina-based recipes are served in the restaurants and hotels. Sweets, ice creams, smoothies, they are served in the hotels and restaurants. This mufali laddu, these spirulina noodles, spirulina papad, this is, yes, go on, continue. This liquid spirulina food bars, this is US based. And you search on Google, many spirulina based recipes are there. This spirulina based Bangkok, in a hotel of Bangkok is served. Now I come to another point. We had a tough time during Corona crisis. And it was a question, can spirulina help in prevention and treatment of Corona, COVID-19? So we have tried to make a literature survey and we got several evidence-based research publications that Corona can be defeated by using spirulina-based formulations. How to fight coronavirus? Because no effective vaccine and therapy are available. So social distancing, isolation, hand wash, this and that. So next slide, please. This spirulina inhibits H1N1 virus was the study from Japan. Then this figure shows the antiviral activity of spirulina. Next. पानी में उगने वाला सुपर फूड जो इम्यूनिटी बूस्टर के साथ एंटीवायरल भी है. Corona, the spirulina can also be used to produce algal vaccine against corona and other viruses. Next. Yes, corona can be helpful in fight against virus, and so Ramdev Baba was not behind. He has also launched spirulina tablets, and he has blended spirulina with some other herbs, and spirulina tablets are available with Baba. And Baba also told that spirulina can defeat corona. Next, spirulina is most promising agent immunity boosting superfood. Next slide, please. Yes. Now I come to another aspect. Spirulina, different usage of spirulina. It is best food or food supplement, highly nutritious and healthy. It is best such food around the world. There is no better superfood available in the world than the so I am able to see the presentation. So spirulina farming is a kind of farming like other land farming, but it is a perfect eco-friendly model because the theme of this present workshop is sustainable and climate resilience. So we need to discover, we need to use green technologies which favor environmental conservation. So Spina plays a significant role in this, and many developments, recent developments in Spina they are incredible. If you know the details, you know what is this? This US car. It is spirulina-based car, algae-based car, and the algae is cultured in this car itself. It uses sunlight as energy. Biomass is created, and this biomass is converted into fuel within the car, and it car runs. And this थोड़ा सा इसको these algae can be converted, algae can be used as a biofertilizer, it can be used as human food, it can be used as animal food, it can be used as a fish food, it can be used poultry food, and we ourselves have done some work not only on human subjects but also animals. In this conference, Dr. Rajesh Kumar is there. With the help of Dr. Rajesh Kumar, we used spirulina as a food, as an ingredient of food for honeybees. Because during dearth period, we have to give supplementary protein food to the honeybees. And when we add spirulina in their artificial food, performance of colony was far, far better. Similarly, we are also doing vermicomposting, and a small amount of spirulina water is, if sprinkled on the bed, the performance of earthworms becomes far, far better. Similarly, we were maintaining a number of fish aquarium, and we prepare spirulina-based fish food, and the growth of fish was much significantly higher, and their coloration, they were brighter and better than on the conventional food. And if you search on Google, lot of fish food formulations of spirulina are available in the market. So, conclusion of my lecture is spirulina is the best nutritional supplement of the world. It helps in maintenance of good health and in prevention and treatment of diseases. Recent developments in spirulina-based technologies are incredible and unbelievable. If you know details of them, you may see that you may observe that you are reading science fiction movie or science fiction stories. Rooftop harvesting technology has been developed. You can harvest sunlight by culturing spirulina on rooftop. Like solar panels are there, you can put glass panels on your roof and culture spirulina. Then bioreactor technology is there, photobioreactor. Indoor, inside the house, you can culture spirulina. Then algal lamps are there. Green algal lamps have been developed which can use sunlight as a source of energy, culture spirulina, and they same spirulina will give light in the night. You can illuminate your houses with the, even protocols have been developed, prototypes have been developed. You can lighten streets with spirulina-based lamps, and such studies are being done in a number of places differently. For example, spirulina-based lighting system, two groups are working and both are giving very good results. And then green building, spirulina powered building has been developed in Hamburg, Germany. So I say the Spurna promises you not only good health for you, good health for the generations to come. It ensures food security because Spurna is like a land farming, but as compared to other type of farming, it utilizes lesser resources, less space is required, less land is required, less manpower is required, less inputs are required, and outputs are much, much better. So we have to do friendship with our health. Health के साथ दोस्ती is better future of for human society. Healthy and energy, one should. Spirulina can restore not only our health, but it can restore our planet also. हमारी धरती माँ ग्लोबल वार्मिंग के बुखार से तप रही है और हमको उस धरती माँ को ग्लोबल वार्मिंग के बुखार से बचाने के लिए हमें ग्रीन टेक्नोलॉजी की आवश्यकता है और स्पिरुलिना टेक्नोलॉजी में वह दम है कि हम अपनी माँ को ग्लोबल वार्मिंग के बुखार से बचा सकते हैं। तो मैं समझता हूँ कि इतना काफ़ी है और thank you very much for giving me opportunity to talk to you. And I believe before I started, he was also talking on spirulina. I don't know what was there. Anyway, thank you very much.
Thank you, sir, for highlighting the importance of the superfood biodata. Even though it's not very popular, but it's a rich source of protein and it's a good investigator. Thank you so much, sir. Your expertise has added great value to this talk. Thank you. Before we proceed further, there's an important announcement for all the participants and community members. Kindly collect your lunch coupons from the registration. Kindly collect your lunch coupons from the registration. Not right now. There's another session. A true leader has the confidence to stand alone, the courage to make tough decisions, and listen to the needs of others. This is a moment of pride and honor for me that we have such distinguished personalities amongst us who enlighten knowledge, biology, and Welcome, sir. And this moment has been graced by Dr. Parmeet Kaur, who is Professor and Head Pathology, Government Medical College, Amritsar. I extend welcome to you.
Good afternoon, everybody. I am really thankful to the management of Baba Park Singh University who invited me for this guest lecture, and special thanks to the Honorable Worthy Vice Chancellor Sir, Dr. Taranjeet Singh. I am basically from a medical background, so I will try that the things which uh, I deliver uh, are easily understood by all of you. Colorectal cancer and role of microbiome. So this was a project which was jointly done by me and my better half, Dr. Purmeet Kaur. He is professor and head in the Department of Pathology in Amritsar. Next slide, please. So what is basically colorectal cancer? It affects the colon and the rectum. That is the large intestine. We know that we have gastrointestinal system, and it starts from the oral cavity, from the mouth, and ends at anus. So the larger part that is the large intestine. The normal cells in this large intestine, which is basically of colon and rectum, is the lining of colon or rectum. It begins to change, and they start uncontrolled replication. It may begin as a non-cancerous polyp, colorectal adenoma, and then grows with time to become cancerous over time. So our intestine is lined by epithelial cells. These are single cells which start changing. So from a single cell, it becomes a polyp. From polyp, then it becomes a growth. And this growth is uncontrolled. After, you know that when our cells divide, there is a controlled division of cells. This controlled division is lost, and this controlled division of cell is lost, and this uncontrolled division leads to the formation of cancer. Next. This is a diagrammatic representation of the large intestine. It starts from cecum. Can I have that? Okay, point. It starts from cecum, then next is the colon, then rectum, and finally the anus. Uh, this is the percentage, like in cecum, the chances of this colorectal cancer is 8%, then in transverse colon 6%, descending colon 10%, sigmoid colon 20%, rectum 38%, and anus 3%. So you can see the last part of our large intestine, that is the sigmoid colon and rectum. It accounts for 58% of the total number of cancer. Next. This is the third most common cancer in the world among both the genders and accounts for 2 million new cases worldwide. And annually, about 1 million people they die of this colorectal cancer. You can see the mortality rate in this. It is a very high mortality rate. from 2 million new cases, 1 million people they die annually. And the number is increasing day by day due to our changing food habits. Next. Symptoms associated with CRC. As we move from left part of the intestine to the from right to the left side, the symptoms relatively change. Initially, there is pain and mass with rectal bleeding. So, if there is pain in the abdomen, along with the feeling of mass over there, plus there is bleeding in the stool. So, that is an indication of presence of the cancer. So, bleeding has many reasons. One is dysentery. One is piles. So, but if there is bleeding, it should be thoroughly investigated. If you have bleeding in the stool, it should be thoroughly investigated. And if it is associated with pain and feeling of mass, it indicates that a cancer is developing in the right side of the large intestine. Then if we move from right to the left side, you can see below the rectum, here the chances of bleeding are 60%, along with change in the bowel habit. Change in bowel habit means sometimes you have constipation, sometimes you have diarrhea. So if you have bleeding along with diarrhea and constipation going alongside, it is an indication that a cancer may be developing in the left side of the large intestine. Next. These are various stages of cancer that initially it is within the innermost layers of the cells, the intestine. Next slide. So here you can see stage initially it is within the intestine, then it goes outside the intestine, involve the lymph nodes, and then involve the liver and lungs finally. That is the stage 4. Next. What are the risk factors for development of this type of cancer? Age, diet, inflammatory bowel disease, family history, sedentary lifestyle, alcohol, smoking, diabetes, obesity, and gut microbiota. So on age, we have no control. It's going to increase with time. Diet, we can control it. We need a healthy diet for our healthy gut. Inflammation in the intestine also associated with diet and various diseases of the gut. Then family history, sedentary lifestyle, diabetes, obesity, alcohol, smoke. So all these are associated with the last point, that is the gut microbiota. That is the bacteria and yeast which are present in our large intestine. There is a word called gut feeling. ਹੁੰਦਾ है ना कई कहते gut feeling. तो gut feeling क्यों किया जाता है? Because microbes which are present in the gut is our intestine. So they release certain chemicals, and these chemicals they are associated with our brain. So our feelings they are also due to these chemicals. So that is why we call it gut feeling. So this is a very important part. Our body has around 37 trillion cells, but we have more than 100 trillion bacteria. The number of cells which a human body has and the number of bacteria which are present in our body, the ratio is 1 is to 10. That is, there are for every 10 bacteria, we have one human cell. The number is so much. And if we weigh them, that is 3 kg. The gut biome or microbiome, that is the bacteria which are present in our large intestine, are so important. Next. This is a genetics part associated with the colorectal cancer. So there are 67 genes which are responsible for maintaining the uh, harmony inside the intestine. And there are approximately 13,000 mutations which are responsible for this cancer. Next. Next slide. In our body, we have oncogenes and anti-oncogenes. Oncogenes are the genes which are responsible for producing a cancer. And we also have anti-oncogenes which suppress these genes. So there is always a balance which is maintained. Next slide. So what is the role of these bacteria in development of cancer? So more than 100 trillion microorganisms they reside in the intestinal tract and play an extremely important role in the human health. These microbes they maintain the intestinal homeostasis by regulating various biological activities. Now, what these bacteria do? What is the role of these bacteria in the intestine? So they break down the drugs and toxins. Our body continuously producing toxins. We are taking drugs. So all these things they are broken down by these bacteria. They do perform many metabolic functions. They regulate our immune response. They protect us against infections and inflammations and many immunological disorders. So when they break down the drugs and toxins, they produce certain chemicals. When the bacteria they change, that is our gut microbiome changes, it leads to formation of different chemicals. And that is the crux, that is basically the reason of development of inflammations and cancer. Next. We call this word as dysbiosis, or in easy terms, it is disequilibrium in the GIT microbiome, which causes various intestinal diseases. Inflammation due to infection of cancer causing bacteria is a well recognized factor. Now, how these bacteria they cause the cancer? Cancer is what is cancer? It is basically uncontrolled growth. Uncontrolled growth. So that is the control on the growth of cells is lost. How it is lost? Bacteria, while breaking down the various chemicals, various drugs, they release toxins. These toxins they damage the DNA due to their oxidative stress. When the DNA is damaged, if the damage is in the part of the DNA which is responsible for controlling the replication of the cells, this will lead to uncontrolled replication. fibrous diet, it produces chemicals which is immune response. So that is why the fibrous diet is so important. Next, next. These are different bacteria which are responsible for these things. Streptococcus bovis, enterotoxic, Fusobacterium, E. coli, Streptococcus, etc. Our intestine has 99% of anaerobic bacteria and very few facultative. The meaning is that these bacteria they grow in anaerobic environment, that is in the absence or less presence of oxygen. So they require oxygen deficient environment for their growth, and these are the good bacteria. Next, so dietary factors which are important for a good gut or a good health are high consumption. These are the consumption of red meat. So consumption of red meat should be minimized. I told you that if a high fat diet leads to production of toxin, so high consumption of red produces toxin. Animal and saturated fat produces toxin. Alcohol, smoking, refined carbohydrates, low consumption of whole grains, low fiber diet, and less fruits, less green leafy vegetables. All these things ultimately lead to production of colorectal cancer. Next, so to reduce the chances of development of this cancer, we can improve our diet by increasing the fibrous diet, reducing the red meat, reducing the animal fats, by increasing the consumption of fruits and green leafy vegetables. This will ultimately produce a good microbiome, and this will enhance the immune response in our gut, and a better immune response will reduce inflammation, and this reduction in inflammation ultimately reduces the chances of development of cancer. Next, when we change our food, our bacteria they also change. So this new these changed bacteria, if we can recognize them, then we can predict the development of cancers in the future. You understood? When we change our diet, our bacteria they change. If we detect this change at an early stage, we can predict the development of cancers in the next 8 to 10 years. So there is a high chance or high, you can say, uh, in future, if you develop these tests or you can detect these changes, this development of cancer can be reduced. If it is predicted or diagnosed at an early stage, so you can treat it at an early stage. And the treatment of cancer, if it is done at an early stage, you can cure it. Otherwise, once it involves the lymph nodes and then liver and lungs, there is no chance of cure. Next, we can screen these bacteria from the human stool or from the human cancerous tissue. Next, for prevention and treatment of this cancer, dietary intervention, probiotics use, prebiotics, symbiotics, and another term is fecal microbiota transplantation. You have heard of kidney transplant, heart transplant. So there is another term which is done by gastroenterologists, that is fecal microbiota transplantation. So it is commonly used nowadays for various infections. There is one infection called Clostridium difficile. In that, the cure of this disease is only through this method. The fecal matter from a healthy gut is taken and that is transplanted in the diseased person, and the disease is cured. Next, dietary interventions encourages the growth of certain bacterial strains that may convert indigestible dietary components into beneficial metabolites for the host. Healthy diet includes high fiber diet, fruits, vegetables, whole grains, fish, white meat, and soya derivatives. Next, what is a probiotic and what is prebiotics? Probiotic is live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. ਅਸੀਂ ਦਹੀਂ ਖਾਂਦੇ ਆ ਨਾ, ਦਹੀਂ ਕੀ ਹੈ? ਦਹੀਂ ਕੀ ਹੈ? This is lactobacilli. ਕੀ ਹੁੰਦਾ ਹੈ? ਦੁੱਧ ਜਿਹੜਾ ਅਸੀਂ ਦੁੱਧ ਨੂੰ ਜਮਾਉਂਦੇ ਆ ਨਾ, ਉਹਦੇ ਵਿੱਚ ਕੀ ਪਾ ਦਿੰਦੇ ਆ? ਥੋੜੀ ਜਿਹੀ ਦਹੀਂ ਪਾਉਂਦੇ ਆ, ਹੈ ਨਾ? ਉਹ ਦਹੀਂ ਦੇ ਵਿੱਚ ਕੀ ਹੈ? ਬੈਕਟੀਰੀਆ ਹੈ. So that is basically we are culturing the bacteria inside the milk. So ਦੁੱਧ ਦੇ ਵਿੱਚ ਬੈਕਟੀਰੀਆ ਐਡ ਕਰ ਦਿੰਦੇ ਆ, ਬੈਕਟੀਰੀਆ ਗਰੋ ਕਰਦੇ ਆ, they release certain chemicals. ਉਹ chemicals ਜਿਹੜੇ ਕੀ ਹੈ? ਪ੍ਰੋਟੀਨਸ ਨੂੰ coagulate ਕਰ ਦਿੰਦੇ ਆ, which are present in the milk. ਉਹ coagulation is ਕਿ ਦਹੀਂ ਜੰਮ ਗਿਆ, ਦੁੱਧ ਜੰਮ ਜਾਂਦਾ, ਮਤਲਬ ਦਹੀਂ ਜੰਮ ਗਿਆ. That is just like a probiotic. So ऐसे तरह there are capsules, tablets, they are available as probiotics. Prebiotic ਕੀ ਹੁੰਦਾ? Probiotic bacteria जो खांदे है, that is prebiotic. तो ਉਹ जी चीज कोई nutritional supplement ਜਿਹੜਾ ਤੁਸੀਂ ਦੇ ਦਿਓ ਜਿਹਨੂੰ probiotic bacteria they eat and they grow, that is a prebiotic. So all these things are basically changing. एक ना acidity बड़ी common term है ना, acidity सारे acid peptic disease भी common है. तो basically वो क्या ਜਾਂਦਾ ਸੀ ਕਿ pH ਘਟਣ ਦੇ ਨਾਲ जो pH acid environment is pH when reduces, now the things are changing. वो कहते pH ਦੀ ਜਗ੍ਹਾ ਤੇ HP है ना, HP is a bacteria. ਕਿਹੜਾ bacteria? That is called Helicobacter pylori. ਕਹਿੰਦੇ ਐਕਚੁਅਲ ਚ ਰੀਜ਼ਨ ਕੀ ਹੈ? pH ਘਟਣ ਦਾ ਕਾਰਨ ਕੀ ਹੈ? That is HP. So Helicobacter pylori. तो ਉਹਦੀ ਪ੍ਰੈਜ਼ੈਂਸ ਜਿਹੜੀ ਹੈ ਉਹ ਰਿਡਿਊਸ ਕਰ ਦਿੰਦੀ ਹੈ pH. ਉਹ pH ਰਿਡਿਊਸ ਹੋਣ ਦੇ ਨਾਲ ਕੀ ਹੋ ਰਿਹਾ ਹੈ? We have acidity and ulcers. So if we our diet, if we our gut microbiome will improve, and once it improves, this leads to healthy habits, healthy changes in our lifestyle. आजकल ਅਸੀਂ जो कुछ भी खा रहे हैं basically that is saturated and fats and refined sugars. Plus जिस तरह मैदा हो गया, हो गया, all these things they lead to acidic environment in our body, acidity and a bad microbiome in the gut. So this is what I want to say, and thank you very much for your you for your patient listening. And I know it's lunchtime now, and thank you very much. If you have any question, you can ask. Thanks a lot.
Thank you so much, sir. It was an insightful and enlightening. You have rightly conveyed that how our diet has adverse effect on the development of colon cancer, and alterations in the diet and proper diet management can alter the colonization of the microbiota. And the healthy microbiome is the best remedy from this cancer. Thank you so much, sir. So, I would like to add that our chief guest, Dr. Daisy R. Batish, from the Department of Botany, University, Chandigarh, she has joined us, and I would like to extend cordial welcome to her. Thank you so much, ma'am, for joining. Now, let's move to the session of honoring our guests of honor. I would like to request our respected Vice Chancellor, sir, along with the conveners, to please honor our guest of honor. Dr. Kamardeep Please welcome Dr. Vic on the stage with a round of applause, all of you. Our honorable guest, Dr. Kamardeep Singh. guest of honor, Dr. Daisy. Thank you so much, all of you. Now we will proceed for the lunch break. As I've already told, please collect your lunch coupons from the registration committee. All are requested to gather here in the seminar hall after the lunch for the post-lunch session. First 20 minutes, the participants and students will go for lunch. After 20 minutes, the faculties follow. Thank you. Twenty minutes, the participants and students will go for lunch. After 20 minutes, the faculties follow. Thank you.
Scientist. First of all, I would like to say that a topic chosen for the conference, that is empowering, first of all, bringing science, technology, and climate, a topic chosen for the conference, that is empowering sustainability. In depth, first of all, the meaning of the technology of the conference, climate, that is empowering sustainability, that is empowering sustainability, that is the dire need of the topic of the conference, climate, that is, and this empowering sustainability in depth, it cannot be brought about together climate without the combination of science, technology. So science, the technology is dependent on the science, and this climate resilience, it is dependent on the technology. I think you are aware the meaning of this मिलेगा तो आराम ਦੇ ਨਾਲ So I am very happy that I have been invited in this university as a guest to address the students, eminent scientists. First of all, I would like to say that the topic chosen for the conference, that is empowering sustainability, bringing science, technology, and climate resilience. Now, if you see in depth the meaning of the topic of the conference, that is empowering sustainability. So, to empower sustainability, that is the dire need of the present days. And this empowering sustainability, it cannot be brought about together without the combination of science, technology. So, science, the technology is dependent on the science, and this climate resilience, it is dependent on the technology. I think you are aware the meaning of this resilience. One who can bear everything, every adverse thing can be feared by anyone. That is the resilient or resilience. Our environment, you know, our climate is bearing all the adverse conditions which have been made by the human disturbance. So the topic is very relevant, and it is the need of the present day that this science, technology, climate resilience should be combined with one another so that we can save our environment. So respected Chancellor Sir, my very dear friend, Vice Chancellor Dr. Taramjeet Singh, and when I was in the university, I could see that there are many students who have been taught by me. First, I would like to recall Dr. Purushottam Singh. When I was doing PhD, he was doing MSc at that time. Then Dr. Indu is there. He was my student. Dr. Vikas is there. Dr. Vikrant is there. So I am very happy to see them all rising. So coming to the talk, that is the climate resilience. How science and technology is responsible for this resilience. Can I have some light here? Anyhow, no. Thank you. So coming to the topic, in the recent time, the human activities are fast disturbing the natural ecosystem. As I have just told you, that our climate is bearing all the disturbances. And the main cause for these disturbances is the human activities. Whatever the activities are there, whatever the fertilizer we are using, whatever the fossil fuels we are consuming, or whatever the pesticides, fungicides we are spraying on the crops, all these are responsible for this climate change. So the climate change, they are putting stress on the agricultural crops. Every crop, has to be sprayed with the pesticide, fungicides, insecticides. The important climate-based abiotic stresses in the plants are like drought or water scarcity. Now, due to this climate change, all these crops, they are facing the water scarcity, salinity, then excessive heat in the summer and cold in the winter. Now you can see nowadays excessive cold is there. In the summer season, you will see that excessive hot season is there. Unbearable temperature in the summer season is there. And so the crops, they are also unable to bear all these climatic changes. It is a challenge to science and technology to develop climate resilient crops which can fight drought, heat, cold, and the soil toxin. Now, the plants are the crops. They are living being like us. As we are breathing, they are also breathing. As we are uh, doing the metabolic activities, they are all doing the metabolic activity, and they need the congenial environment. But due to the human activities, this congenial environment that is not available to our crops. That is why the scientists, they are searching and doing regular researches so that to improve the crops in order to face all these adverse environmental conditions. So the theme of this conference, as I have already told you, has nicely been selected by the organizing secretaries or organizers, bringing science and technology for climate resilience. So all these three topics, science, technology, and climate, as I have already told you, they are dependent on one another. So it is definitely going to benefit the finding out the ways and means to develop the climate adverse climatic resilient crops. The collaboration of biological sciences and technology gave rise to new field of science, biotechnology, which work in this direction. मिलेगा तो आराम दे नाल So I am very happy that I have been invited in this university as a guest to address the students, eminent scientists. First of all, I would like to say that a topic chosen for the conference, that is empowering sustainability, bringing science, technology, and climate resilience. Now, if you see in depth the meaning of the topic of the conference, that is empowering sustainability. So, to empower sustainability, that is the dire need of the present days. And this empowering sustainability, it cannot be brought about together without the combination of science, technology. So science, the technology is dependent on the science, and this climate resilience, it is dependent on the technology. I think you are aware the meaning of this resilience. One who can bear everything, every adverse thing can be feared by anyone. That is the resilient or resilience. Our environment, our climate is bearing all the adverse conditions. which have been made by the human disturbance. So the topic is very relevant and it is the need of the present day that this science, technology, climate resilience should be combined with one another so that we can save our environment. So respected Chancellor Sir, my very dear friend, Vice Chancellor Dr. Taramjeet Singh, and when I was in the university, I could see that there are many students who have been taught by me. First, I would like to recall Dr. Purushottam Singh. When I was doing PhD, he was doing MSc at that time. Then Dr. Indu is there. He was my student. Dr. Vikas is there. Dr. Vikrant is there. So I am very happy to see them all rising. So coming to the talk, that is the climate resilience. How science and technology is responsible for this resilience. Can I have some light here? Anyhow, no. Thank you. So coming to the topic, in the recent time, the human activities are fast disturbing the natural ecosystem. As I have just told you, that our climate is bearing all the disturbances. And the main cause for these disturbances is the human activities. Whatever the activities are there, whatever the fertilizer we are using, whatever the fossil fuels we are consuming, or whatever the pesticides, fungicides we are spraying on the crops, all these are responsible for this climate change. So the climate change, they are putting stress on the agricultural crops. Every crop, has to be sprayed with the pesticide, fungicides, insecticides. The important climate-based abiotic stresses in the plants are like drought or water scarcity. Now, due to this climate change, all these crops, they are facing the water scarcity, salinity, then excessive heat in the summer and cold in the winter. Now you can see nowadays excessive cold is there. In the summer season, you will see that excessive hot season is there. Unbearable temperature in the summer season is there. And so the crops, they are also unable to bear all these climatic changes. It is a challenge to science and technology to develop climate resilient crops which can fight drought, heat, cold, and the soil toxin. Now, the plants or the crops, they are living being like us. As we are breathing, they are also breathing. As we are doing the metabolic activities, they are all doing the metabolic activity and they need the congenial environment. But due to the human activities, this congenial environment that is not available to our crops. That is why the scientists, they are searching and doing regular researches so that to improve the crops in order to face all these adverse environmental conditions. So the theme of this conference, as I have already told you, has nicely been selected by the organizing secretaries or organizers, bringing science and technology for climate resilience. So all these three topics, science, technology, and climate, as I have already told you, they are dependent on one another. So it is definitely going to benefit the finding out the ways and means to develop the climate, adverse climatic resilient crops. The collaboration of biological sciences and technology gave rise to new field of science, biotechnology, which work in this direction. The climatic changes, they are converting our fertile agriculture into non-fertile by increasing the alkalinity, acidity, desertification, then the biological or microorganisms, they play a very important role in the production of our crops, biological microorganisms. So they are being killed by the acidity of the soil, by the alkalinity of the soil, or when we put fertilizers, or when we put these pesticides, insecticides on the crop. So blue-green algae have the potentiality to reduce the acidity and alkalinity and reclamation of the agricultural land to restoring its fertility. So this blue-green algae, it is also the microorganism which is responsible to face the adverse climatic condition in order to increase the production of the crop plants. The agricultural scientists, they are highly concerned regarding climatic changes and making all out efforts to develop resistant crop varieties so as to feed the ever increasing population. When these crops, they have to face the adverse climatic condition, naturally whole of their metabolic system is disturbed. And when the metabolic system of the plant body is concerned, then they are unable to produce more and more. Whereas our population is increasing day by day, and on the other hand, due to changes in the climatic conditions, the crop production is being reduced. So currently, our country is facing a challenge of producing the adequate food to meet the demands of ever increasing population from shrinking natural resources, intensification of agriculture to enhanced productivity, and resource use efficiently has to be the main focus as competition for land and water are increasing from non-farming sectors. Further, in recent years, the climate change and its vulnerability are emerging a major challenges to Indian agriculture. In view of these, immediate thrust is needed on enhanced production with reduced natural resources under a variable climate. So this climate resilience, that is the very, very important burning topic of these days. The development and identification of climatic resistance crop varieties with enhanced tolerance to heat, drought, flooding, killing, salinity stresses, they are very, very essential in order to sustain the improved crop yields to cope up with the challenges of the climatic changes. So it means heat. We have to reduce or we have to reduce the global warming, that is this heating effect on the plants. Then drought, we have to produce the drought resistant plants. Then flooding, flood resistant plants. Last year there were great floods in the Punjab, and whole of the rice crop was destroyed. So we have to face all these situations. As a scientist, I can say that most of the scientists, they are working in these type of the searches so as the plants they become resistant to the climatic changes. That is the climatic resilience. Climatic resilience, as you know, it is the ability to anticipate, prepare for, and respond to hazardous events or disturbances related to the climate. So, once again, I will say that all these disturbances they are due to the human activity. We are responsible. All of us are responsible for this climatic change. Now, improving climatic resilience involves the assessing how climate change will create new or alters the current climatic related risk and taking steps to better cope up with these risks. So, how to protect our crops from all these risks of salinity, drought, acidity, etc. Then, climate resilient crops. They are the crops and the crop varieties that have enhanced tolerance to biotic and abiotic stresses. I think you are familiar with both these terms, biotic and abiotic stresses. In order to create mass awareness for adoption of climatic resilient technology, the Prime Minister of India on 28 September 2021 dedicated 35 crop varieties with special traits via video conferencing. The event was organized at all at the Indian Agricultural Research Institute, New Delhi, ICAR, Indian Council of Agriculture Research. These 35 new crop varieties with special traits such as climatic resilience and nutritional rich contents have been developed by ICAR to address the twin challenges of climatic change and malnutrition. These include a drought tolerance variety of chickpea, wilt, and sterility mosaic resistance pigeon pea, early maturing varieties of soybean, disease resistant varieties of rice, and biofortified varieties of wheat, pearl millet, maize, and chickpea, quinoa, buckwheat, winged bean, and faba bean. These special trait crops varieties, they also include those that address the anti-nutritional factor found in some crops that adversely affect the human and animal health. So in the last, I will say that it is not only the plants which are affected by this climatic change. It is the human being also and all the living organisms, even the smallest living organism, that is the viruses, they are also affected by this climatic change. So our duty is that whatever we learn from this conference, we should apply in our daily life. There are many activities which we can avoid or which we can perform in order to save our environment. So, with this brief introduction, I once again thank the Vice Chancellor and the organizers of this conference who have invited me to this conference. Thank you very much. Thank you.
Thank you so much, sir. Being a plant biochemist, I can feel the pain that is being faced by plants, and of course, our duty to take appropriate efforts to bridge science and technology for empowering sustainability. Thank you so much.
We have been graced by the
मिलेगा तो आराम दे नाल so I am very happy that I have been invited in this university as a guest to address the students, eminent scientists. First of all, I would like to say that the topic chosen for the conference, that is empowering sustainability, bringing science, technology, and climate resilience. Now, if you see in depth the meaning of the topic of the conference, that is empowering sustainability. So, to empower sustainability, that is the dire need of the present days. And this empowering sustainability, it cannot be brought about together without the combination of science, technology. So science, the technology is dependent on the science, and this climate resilience, it is dependent on the technology. I think you are aware the meaning of this resilience. One who can bear everything, every adverse thing can be feared by anyone. That is the resilient or resilience. Our environment, you know, our climate is bearing all the adverse conditions which have been made by the human disturbance. So the topic is very relevant, and it is the need of the present day that this science, technology, climate resilience should be combined with one another so that we can save our environment. So, respected Chancellor Sir, my very dear friend, Vice Chancellor Dr. Taramjeet Singh, and when I was in the university, I could see that there are many students who have been taught by me. First, I would like to recall Dr. Purushottam Singh. When I was doing PhD, he was doing MSc at that time. Then Dr. Indu is there. He was my student. Dr. Vikas is there. Dr. Vikrant is there. So I am very happy to see them all rising. So coming to the talk, that is the climate resilience. How science and technology is responsible for this resilience. Can I have some light here? Anyhow, no. Thank you. So coming to the topic, In the recent time, the human activities are fast disturbing the natural ecosystem. As I have just told you, that our climate is bearing all the disturbances. And the main cause for these disturbances is the human activities. Whatever the activities are there, whatever the fertilizer we are using, whatever the fossil fuels we are consuming, or whatever the pesticides, fungicides we are spraying on the crops, all these are responsible for this climate change. Has to be sprayed with the pesticide, fungicides, insecticides. The important climate-based abiotic stresses in the plants are like drought or water scarcity. Now, due to this climate change, all these crops, they are facing the water scarcity, salinity, then excessive heat in the summer and cold in the winter. Now you can see nowadays excessive cold is there. In the summer season, you will see that excessive hot season is there. Unbearable temperature in the summer season is there. And so the crops, they are also unable to bear all these climatic changes. It is a challenge to science and technology to develop climate resilient crops which can fight drought, heat, cold, and the soil toxin. Now, the plants are the crops. They are living being like us. As we are breathing, they are also breathing. As we are uh, doing the metabolic activities, they are all doing the metabolic activity, and they need the congenial environment. But due to the human activities, this congenial environment that is not available to our crops. That is why the scientists, they are searching and doing regular researches so that to improve the crops in order to face all these adverse environmental conditions. So the theme of this conference, as I have already told you, has nicely been selected by the organizing secretaries or organizers, bringing science and technology for climate resilience. So all these three topics, science, technology, and climate, as I have already told you, they are dependent on one another. So it is definitely going to benefit the finding out the ways and means to develop the climate adverse climatic resilient crops. The collaboration of biological sciences and technology gave rise to new field of science, biotechnology, which work in this direction. The climatic changes, they are converting our fertile agriculture into non-fertile by increasing the alkalinity, acidity, desertification, then the biological or microorganisms, they play a very important role in the production of our crops, biological microorganisms. So they are being killed by the acidity of the soil, by the alkalinity of the soil, or when we put fertilizers, or when we put these pesticides, insecticides on the crop. So blue-green algae have the potentiality to reduce the acidity and alkalinity and reclamation of the agricultural land to restoring its fertility. So this blue-green algae, it is also the microorganism which is responsible to face the adverse climatic condition in order to increase the production of the crop plants. The agricultural scientists, they are highly concerned regarding climatic changes and making all out efforts to develop resistant crop varieties so as to feed the ever increasing population. When these crops they have to face the adverse climatic condition, naturally whole of their metabolic system is disturbed. And when the metabolic system of the plant body is concerned, then they are unable to produce more and more. Whereas our population is increasing day by day, and on the other hand, due to changes in the climatic conditions, the crop production is being reduced. So currently, our country is facing a challenge of producing the adequate food to meet the demands of ever increasing population from shrinking natural resources, intensification of agriculture through enhanced productivity, and resource use efficiently has to be the main focus as competition for land and water are increasing from non-farming sectors. Further, in recent years, the climate change and its vulnerability are emerging a major challenges to Indian agriculture. In view of these, immediate thrust is needed on enhanced production with reduced natural resources under a variable climate. So, this climate resilience, that is the very, very important burning topic of these days. The development and identification of climatic resistance crop varieties with enhanced tolerance to heat, drought, flooding, chilling, salinity stresses, they are very, very essential in order to sustain the improved crop yields to cope up with the challenges of the climatic changes. So it means heat. We have to reduce or we have to reduce the global warming, that is this heating effect on the plants. Then drought, we have to produce the drought resistant plants. Then flooding, flood resistant plants. Last year there were great floods in the Punjab and whole of the rice crop was destroyed. So we have to face all these situations. As a scientist, I can say that most of the scientists they are working in these type of the searches so as the plants they become resistant to the climatic changes, that is the climatic resilience. Climatic resilience, as you know, it is the ability to anticipate, prepare for and respond to hazardous events or disturbances related to the climate. So, once again, I will say that all these disturbances they are due to the human activity. We are responsible. All of us are responsible for this climatic change. Now, improving climatic resilience involves the assessing how climate change will create new or alters the current climatic related risk and taking steps to better cope.
knowledge and inventorization of high species. After completing from the Punjab University, she started her career as Punjab University. She had 21 years of teaching, more than 120 research papers, 7 edited, 35 book chapters. She had visited more than 10 countries, attending conferences, delivered 105 lectures, and she had successfully guided 25 PhD students and 10 MPhil students. She remained in many national and international institutions and taught in many abroad. She also remained the chairperson of Department of Botany. Now I request Dr. Daisy to kindly deliver the talk.
Thank you, Dr. Vikrant. Uh, a very good afternoon to all of you. First of all, I express my to the authority, Vice Chancellor, and other dignitaries of the university for giving such a warm welcome and inviting for this important lecture today. So I'm extremely thankful all the college authority, especially Dr. Vikrant, who happens to be our student, and the great satisfaction to see the students going to heights and organizing functions. So I'm thankful to all for inviting for this important. For today's lecture, I chose one of the important environmental problems which is globally, which whole of the world is facing, and this is a problem which I would say double problem. On one side, we are facing changing climate, and on the other side, we have such a kind of vegetation which unchecked, which grows uncontrolled, and the control of which is headache for the science. is likely to become endangered, they have to face these same hurdles, just same challenges, same evolutionary challenges like the demographic bottlenecks, like hybridization, like new environment. So endangered species also enters into new environment because the environment becomes problematic for that. So it is new for endangered species. Likewise, invasive species which has come from altogether new environment, the other environment is also new for it, and then they have to face the demographic bottlenecks, genetic changes it has to face. So both, one is declining, other is expanding. So both must face the same evolutionary bottlenecks. So that is very ironic that endangered species and the these invasive species, they have something common in them. Next, please. Yeah. So just briefly, I would say that invasive species are intentionally brought because some are very beautiful, some are very productive, some are good for agriculture, some are good for forestry. So we sometimes intentionally bring them, but some of the invasive species, they also come unintentionally. They also come accidentally in the other nations, and they have to then bear them. For example, Parthenium hysterochorus, well known as Congress grass, is unintentional introduction in not only in our country but in various other countries. And ageratum, lantana, they have been brought purposely for the ornamental value because of their ornamental value. Next, please. Yeah. So why we are talking of invasive species and why, what do they do in our environment? One is that they cause biodiversity loss. They are the second major cause of biodiversity loss. They cause species extinction, and they are because they interfere with crops. They also have effect on food security and the livelihood. So livelihood of the people are also affected. So this is one of the social, effect of the impact of the invasive species. Then they are significant drivers of environmental degradation. Wherever invasive species are present, environment becomes degraded in those areas. Then, because they are threat to biodiversity, so they lead to ecosystem loss of ecosystem integrity, and they also cause huge economic costs. Economic cost, one, in terms of the loss they cause. Second, when we want to correct that problem, we have to spend even more money. So, as per a study in 2019, US dollar 423 billion, they were, the economic loss faced due to invasive species only. So you can imagine huge economic loss one has to face because of the presence of invasive species. Yeah. So these are some of the native species of Himalayas which are endemic to Himalayas, and they are gradually being replaced by the exotic species, by the invasive species. Now one is Dictylorhiza, the second one is Boschianica, and third is Arnica. So these are some rare medicinally important plants of Himalayas which are becoming endangered because of the entry of invasive species in those areas. Next, please. You can see the structure of the ecosystem gets altered. This is Lantana tamara, which is present in the lower hilly tracts of Himalayas. You see that earlier we used to have other herbs and shrubs, which were probably important to the native people, but nowadays Lantana has replaced them. And lantana, the fruit of lantana, it is poisonous, and many people are eating them because of the unawareness, because they are not aware that it is poisonous, and they are eating it. We asked many students in school students in Himachal, and they say that on their way when they come to school they eat it. So this kind of practice is going on in many areas. So this is because of the lack of knowledge
that people are doing this. They are eating its fruits, its berries, which are very poisonous. Although the level of poison may vary from species to species, from area to area, but it is not good for health. So it alters the composition of native communities. Next, please. Now just imagine these are the four plants I have used. I have selected here. Parthenium is there. Ageratina is there. One is Hyptis. Hyptis is Vilaiti tulsi. Now, this plant is otherwise used to be medicinally important, but nowadays it is becoming invasive not only in northern part of India, but also in many areas of south, northeast area, particularly in Uttar Pradesh, Vindhyan Hills. They are the worst sufferers because of this heat. Next, please. Now, when these weeds spread, so there is one mechanism, allelopathy. These weeds, one is they mechanically do not let any other plant to come in their vicinity. Second is that they release chemicals through their roots, through their leaves, and also through their dead and decaying parts. They release chemicals, and those chemicals they are toxic for other plants. They are not toxic to the donor plant, but they are toxic to the other plants, which is a phenomena known as allelopathy. So allelopathy is one phenomena that is responsible for the unchecked growth of these weeds. So it's very important that because of the allelopathy property, a very important hypothesis is there, which is known as novel weapon hypothesis. Now, people say that these chemicals serve as novel weapons for other plants, for native plants. They are weapons, and they are using chemicals as weapons to kill the surrounding vegetation. And this is what novel weapon hypothesis, which is related to invasive species, and which is one of the cause or mechanism of spread of invasive species. Next, please. Yeah. So they disrupt ecosystem, they reduce biological integrity, the pristine environment of Himalayas, of urban environment, or vacant areas. Where earlier we used to have the important forages and herbs. They have all been replaced by the invasive species, and they reduce biological integrity. Next, please. Now these affect major ecosystem services. These are some of the plants that are normally found in these areas. They have now been replaced by the species. Now, for example, Abutiloniticum, for example, Adatuna. Adatuna is traditionally important. Those from botany they know its importance. And the Anisomylis, it is one of the aromatic plants. Barlaria is one of the very important medicinal plants of Himachal Pradesh. Then Carissa carenta is Carissa edible, and then Dioscorea, it is also a medicinally important plant. Buraya kiwigiai, I think you all know. Buraya kiwigiai is very good. It's so important. It's seen widely. That's the lily of season. This is a water crop. So all these they have become reduced because of the presence of invasive species. Their abundance, their density has declined, and in certain areas these species are missing altogether. So they cause fodder scarcity. So fodder scarcity means the animals or the livestock, they do not have any grass to eat, but they all find these partheniums or these lantana, ageratum all around, and therefore fodder scarcity is very badly felt by the local people. Next, please. Yeah, this is one of the pictures showing the natural landscapes that they are losing their luster because their understory is full of only some invasive species. They are forming the understory. Earlier the understories used to be very rich with lot of species diversity. Some are flowering, some are patchy, some are just the herbs. So they gave a very good look but when an invasive species enters, it spoils the natural beauty of the landscape. Next please. Yeah, this is one of the slides which holistically show how the ecosystems change when invasive species enters into environment. Just see here that before invasion, ecosystem was very balanced. There was complex food webs. There were going on the complex species interactions. There were natural species assemblages were present. Diverse ecosystems were there. Small habitats were there. And after invasion, the picture changes. Monocultures of certain species can be seen. The altered species interactions are there, and then the food webs, they also become different. They are no longer the natural food webs which we used to have, and then soil microbiota also changes. Then there are changes in the chemistry of soil, and they also affect the food webs. So in this way, the whole ecosystem integrity is somewhere affected because of the entry of the invasive species. So that is one of the very important impact that ecosystem integrity is affected when the invasive species enter into the natural ecosystems. Other impacts can be seen like they affect the cultures that because there are many cultures related with certain native species. If those native species are replaced by invasive species, so those cultures are also affected. Then there are societal impacts. Society has to face water scarcity, food scarcity at certain places, and also scarcity of medicinal plants, scarcity of natural forages. So these, societal changes can also be seen, and then livestock suffer, as I have already told you, loss of genetic diversity. When biodiversity loss is there, genetic diversity is also very badly affected. Then there are health issues related to certain plants. Parthenium, for example, it is health hazardous. It causes allergy to human beings. It also causes rhinitis. It also causes asthma. Many people they are affected. I know the Chandigarh cases. Lot of parthenium used to grow in Chandigarh. People were suffering skin allergies. One of the during when I was doing PhD on parthenium, one of the there used to come one gentleman whose wife was suffering from the skin allergy due to parthenium, and then doctors advised her to go to a place where there is no parthenium. They went to Kasoli, I remember, but nowadays Kasoli is also having a lot of parthenium. So that is the change we see. But now people are aware, so they take care. They avoid touching parthenium, or they don't go into the monocultures of parthenium. Likewise, agitator, which is known as Neela in the bottles, and this is so bad smelling that one feels giddy, one feels headache when it passes through. So there are many health problems which are linked with the invasive plants. They also affect the economy of the nation. They cause food scarcity, etc. And then also in certain places tourism is also affected because beauty of the natural landscape is affected. Obviously, tourism will also be affected. So next please. So these are the same things, but in aquatic environment, when I was coming here, I saw certain rivulets in your part also. They were full of water hyacinth, full of water hyacinth. So when invasive species are present in water bodies, for example, water hyacinth, what they cause? They destroy biodiversity of the aquatic system. They cause oxygen depletion. They become a breeding ground for pests and vectors. They block waterways, hamper agriculture, fisheries, recreational activities, and even hydrocarbons. Next, please. Now, just see this is one picture on way to Batinda. So one of the rivulets was fully, it was full of this water hyacinth. Water hyacinth is very destructive. When it enters into water bodies, within 12 days, it encroaches the whole of the water body. And then it spoils the water quality, it becomes unfit for drinking, and also it becomes useless for the body. even the animals which used to come to be there for drinking water, that is also not available for them because one of the water body gets choked with this. And as one example in Lake Victoria in Eastern Africa, it grew to such a huge densities that even the ships they were unable to leave the docks. So this is a very harmful weed, and we need to recover or restore our water bodies because I've seen in Punjab, Haryana, or on our way to Delhi, all the water bodies are choked. I don't know why this is happening. This weed is spreading at a very fast rate, and we need to do something about it. Next, please. Yeah. Now, something about climate. Climate is changing. We are seeing warming season, or not only warming, unpredictable, weather. Sometimes when it is the weather of rain, rains will come, they come late, or when even the winter season starts late, and when it comes, the intense winter is there, maybe for 2 days, but it is very intense. Likewise, the hot seasons are also very intense. So we are feeling somewhere that climate we can't expect sometimes, but it is true. Everybody says that yes, climate is changing. So next, please. Now this changing climate is giving an advantage to the invasive. These invasive species are spreading. They're so adaptable. They are already adaptable. So changing climate does not affect them. They, grow unchecked, while the other species they bear the brunt of the changing climate. But these invasive species they are growing unchecked because of the climate change. So the resilience, what is resilience? Means ability to come back to normal form. That is also declining because of the presence of invasive species in the natural ecosystems and in the urban environment, in the agricultural environment. So the changing climate is providing more opportunity to invasive species to spread further. So it is causing double headache for us. Not only we are to think about the changing climate, but we are also to think about how why invasive species are also increasing. Next, please. Yeah, it is. I already said because of the changing climate, we have hurricanes, floods, droughts, and invasive species. They are become even more spreading type under such situations. Next, please. Yeah, these are some of the climate change and invasive species how they spread. No, one is that when climate change occurs, they find new ways to spread. They have new pathways to spread, and then their distribution is. Distribution means there are some species which can grow only in hot environment, and now even the cold environment are also becoming hot. So their distribution level increases, and even fabrics are less effective when climate change is there, and invasives emerge earlier and stay for longer time in a particular environment. So I'll discuss these one by one. Next, please. Yeah. Alternate transport mechanisms for the invasive species. For example, Arctic shipping passages because ice is melting in the event of hot environment. So the ships they find alternate because earlier they had to face the ice there, but ice is now melting, so alternate pathways are there for the ships, and these ships are the major pathway for spread of invasive species. So many invasive species they also come along with the new pathways of the ships. Next, please. Yeah. So when climate change, ice cover melts. So there is more water, more pathways, change the pattern of stream flow, warmer water, and also water becomes salinized. Now all these conditions favor certain invasive species to move to the new environment. Yeah. This is hitchhiking. there are certain parts of the ship where the seeds of certain species they stick. This is what we call hitchhiking. So hitchhiking patterns also increase. And now, as per a study, seeds of 30 plant taxa were collected from air intake grill of a refrigerated shipping container. Shipping containers are there were 30 species. They were attached to that container, and this is how they move from one place to another. Next, please. Yeah. Plastics have even, aggravated the problem. It is said that oceans are full of plastics. You must have read the news that in the bed of ocean or even in between oceans, a lot of plastic bottles are there. Now, when invasive, there are many invasive species which are moving from one place to another place through plastic. Even microplastic is also facilitating the movement of many invasive species. So through water currents, they move from one place to another. So the plastics have added agony to the fire. So they have further spread the spread of invasive species. Next, please. Yeah, this is ballast water. Ballast water is used in ships for balancing purpose, or when the ship is moving, moving through the some area. So if it has to maintain balance, ballast water is either added or it is removed from the ship. So along with that ballast water also very invasive are able to dealing with that. Next, please. This is very interesting that on one side we are having quarantine measures, strict quarantine measures that no new species should enter our environment. But then there is online shopping. There are many invasive species seeds which are available online. In Amazon, you can see the seeds of saturatum. You can see the seeds of Lantera. You can see the seeds of even Isonia, the water hyacinth, because this flower is also very beautiful. So these are available online and people are buying them. They are buying these invasive species for beautification. So where is the check? So online shopping or purchases online are also becoming risky, and it is allowing many invasive species to enter into the web. Next, please. Yeah, you can see the picture. So beautiful, this is the datum. So beautiful antenna. The seeds are available online, and people are purchasing them. So there is no point of we are having quarantine measures. We are having so many other measures so that no new species come from the other environments. But online checking is doing this. Next, please. Next. Yeah, wildfires. Wildfires, you now in the television you can see there are fires, in European countries, there are fires in, uh, American countries. So these wildfires, they're making soil and right? They're altering the chemistry of the soil, and these altered soils they are very favorable for invasive. So they are also wildfires are also contributing to the spread of invasive species. This is again very interesting. People have linked this is a study from WII, they are India. They say that decline in the tiger population may also be related to this. This is because they have to eat the herbivores. Herbivores do not find food in the event of invasive species, Lantana, for example, and in this way, because of food scarcity, the top predator tiger also suffers. Next, please. So next, please. Yeah. So this is again interesting that plants are under stress because of the climate change. Under stress, they make DNA. There is methylation of DNA. Histones get proteins. They get modified, and non-coding RNAs are synthesized. all these they enhance the adaptability of invasive. Some genes get silenced, some genes are overexpressed, and this enhances the adaptability of plants. And in this way, they spread they spread more and more areas. Next, please. Next. Yeah, these are some of our studies. So I'll not just speak about them. Next, please. I'll just show you that we have worked extensively on Himalayas. Then next, please. Yeah, this is also. I want to tell this is one species. One right? Yeah, this is one species which is known as Burgess. This is found in Punjab, and when we were coming to this Punjab place on the way also on the way the road divider, lot of Burgess this is very beautiful, but it's very harmful at the same time. So we have done some studies on it, how it affects the soil of Punjab, how it affects the native biodiversity of Punjab. So it is very harmful. Try to maintain it in normal density. I'm not saying that eradicate it, because it is beautiful also. Just looks like sunflower, but try to maintain its densities at a level that they are not harmful. Right? So next please. Next. Yeah, this is our study. So because of the positive time, I will not be able to share much with you. Okay, next. Yeah, this is our next. Next, please. Next. Yeah, this is 50. About this plant, I talked to you that it was a medicinal plant, and you can see how it is spreading. So this is also one of the very harmful. And so in the end, I would say that you should all be aware about the invasive plants growing in your vicinity. Try to manage them, try to control them in densities which are at a low level, right? So that they do not harm your native vegetation. Your native vegetation is very precious. It is very important. It has been giving you so many things, so many ecosystem services. Try to preserve that also. I'm not saying you should eradicate these plants. They're not that harmful, but the densities should be at a manageable level, so that they do not affect. Try to feel the agony of native plants. They cannot speak. Okay, so we take action when we directly face something. Try to face their problem. Try to understand their problem also. So try whenever you see invasive plants in your vicinity, try to control them rather than giving them more space to spread. So that is the message for today. So with that I come to an end. And I once again thank the University of College, Dr. Vikram, and many for inviting me to this important event. So thank you.
सब कहाँ है लिंक नेट खोलना है लिंक शेयर
डॉक्टर सुमित को कर देना चाहिए
from starting from the concept of invasive species. And then briefly explain the other processes of invasive species, and then last studies what we have done. एक बात का मुझे वाक्य याद आया कि when we are ये डैम बन रहे थे तो उसमें वहाँ पर डंपिंग साइट बनी डंपिंग साइट के ऊपर बोल रहे हैं यहाँ पर ना वेजिटेशन तो आईपीडी को ये वर्क मिला तो एट दैट टाइम वो स्पीशीज चूज करनी थी तो क्योंकि मैं उसमें सही टर्म पे काम कर रहा था तो हमारे डायरेक्टर ने बोला कि इसको ना लगा दे ये बढ़िया रहेगा तो मैंने स्ट्रिक्टली मना कर दिया मैंने कहा सर ये बिल्कुल भी सही नहीं रहेगा क्योंकि अगर इसको अगर एक बार लगा दिया तो ये वहां से फिर जाएगा तो हमने भी बाद में बड़े तरीके से जब कुछ ट्रांसमिशन किया था कि हर एक एक्चुअल वाइज कौन सी जो चीज करने बनेगी। ये एक बड़ा इंपॉर्टेंट है कि जब हमें प्लांटेशन करनी है कहीं से तो हम किस तरह की प्लांटेशन करनी है सीजन के हिसाब से प्लान करनी है। तो आई एम थैंकफुल टू यू दैट we sign and directly come. Thank you.
Hello, 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 hello.
There is no doubt now that sustainability has moved to the top of the agenda. But still, many concerns related to the cultural concerns related to the agriculture, technology, and science need to be addressed for nature-based solution for sustainable ecosystem. Now, I'd like to welcome Dr.
Sumit Bharadwaj, Assistant Professor, Department of Physics, Chandigarh University, Mohali. Sir has the area of specialization in material sciences, engineering physics, nanotechnology, polymer composite material for dielectric and energy harvesting applications. He has 30 years of teaching and research experience. He has contributed more than 30 research papers in renowned journals. He is actively involved in supervising master's and PhD students. He holds lifetime membership of various scientific societies. So, sir, I welcome you and sir, kindly deliver your talk.
Am I audible, ma'am? Hello, hello. Hello, am I audible, ma'am?
Now I start my online session. So I repeat the name again. Balvinder Kaur. Baljinder Kaur is there. Baljinder Kaur. Then
Screen is visible,
Shivani
ma'am?
Thakur or Suresh Kumar. Savita Devi. Deepika. Ritu Dhiman.
Hello, the screen is visible,
Divya.
ma'am?
Suresh Kumar. Jinender. Sheetal. Suresh. Vishal Mahi. Deepti Gupta. Mamta Verma. Diksha Chaudhary. Namrata. Kamaldeep.
Diksha Chaudhary, yes, ma'am.
Diksha Chaudhary, you have to present.
दीक्षा आप शेयर करो योर स्क्रीन.
Yes, yes, ma'am, sir.
Am I audible, ma'am?
Yes, sir.
Okay, the screen is visible.
Yes, sir.
Good afternoon.
दीक्षा अपने मोबाइल को लैंडस्केप में करो।
Thank you very much for the kind introduction.
Sorry, sir.
लैंडस्केप में करो इसको।
Okay.
Hello. Hello. Now okay, ma'am? Hello.
Hello.
Yes, ma'am,
हाँ
audible?
जी हाँ जी करो स्टार्ट।
Okay, okay, sir.
Am I audible, ma'am?
Chaudhary. Today I present my oral presentation. My title is Ethnobotanical Uses of Medicinal Plant Justicia odorata. Let's my introduction. Justicia odorata is commonly known in English as Malabar nut. Is a medicinal plant native to Asia in West UP, UK, Himachal Pradesh, and Himalayas region.
Am I audible, ma'am? Hello.
FMPAC, substance SKD, and species Adatoda. It is evergreen, gracious shrub 3-6 meter long, large leaves, lanceolate 10 to 20 by 4-8 centimeter, whereas flowers are white or purple in short, dense, auxiliary pedunculates. The leaves of Justicia adatoda contain phytochemicals such as alkaloids, tannins, saponins, phenolics, and flavonoids. The most important is vaccine, a quinazoline alkaloid. This shrub has a number of traditional medicinal uses in Siddha medicine, Ayurveda, homeopathy, and Unani medicine. Justicia datoda is well known in the indigenous system of medicine for its beneficial effects, particularly in bronchitis. The leaves, flowers, fruits, and roots are extensively used for treating cold, cough, whooping cough, and chronic bronchitis and asthma. The 6 different alkaloids from Justicia datoda leaf can be used as an anti-tubercular agent. These alkaloids inhibit activity of anti-FPH, thereby preventing the initial step of fatty acid biosynthesis and can be effective against tuberculosis. Justicia datoda leaves and root extracts used on diabetes-related homological parameters such as blood glucose levels, glucose tolerance, and glycosylated hemoglobin and serum lipid profile on the weight of normal and diabetic rates. Medicinal plants are plants whose extracts can be used directly or chemically for the treatment of different ailments. The use of traditional medicine and medicinal plants in most developing countries as a basis for the maintenance of good health has been widely observed. The villagers have their common remedies for medical treatment by using various plants or plant products present in their vicinity. Therefore, in the recent years, one can notice a global trend in the traditional system of medicines and ethnobotanical studies have become increasingly valuable in the development of systems in different parts of the world. These are ethnobotanical uses. Justicia datoda is well-known plant herb in Ayurvedic and Unani medicine. Justicia datoda leaf extracts. Previous studies report that leaf contain phenols, tannins, alkaloids. These are vaccine, vacinonine, adatodine, saponins, flavonoids, and reducing sugar and vitamin C. The leaves, roots, flower, and bark of this plant have been used in the treatment of cough, cold, asthma, to liquefy sputum as a bronchodilator, bronchitis, and tuberculosis. The different parts of the plant is used in the Indian traditional medicine for the treatment of various diseases like joint pain, lumbar pain, sand spring, cough, eczema, malaria, rheumatism, swelling, and venereal disease. These are the of literature. Gulsazan and Mercer, Jha, Sandeep Dhankar and Raman Kaur, M. Gulfraz, Pujaram Jeeban Kumar Singh and Deepak Huidrom, Dibadin Bose and Someshwar Chatterjee. That's our conclusion. The literature survey revealed that the Justicia dotata has been widely studied for its pharmacological activities and regarded as universal panacea in Ayurvedic medicines, and finds its position as a versatile plant having a wide spectrum of medicinal activities. It can be concluded that Justicia dotata is an important source of many pharmacologically and medicinally important chemicals such as vacinone, vesicolone, and other various useful minor alkaloids. These are references. Thank you so much.
Okay. What is the significance of your research? Hello, Diksha.
Hello, hello.
What do you understand by ethnobotanical?
Sorry, ma'am.
Ethnobotanicals क्या होते हैं? What do you understand by this term?
Ethnobotanical means.
Yeah.
मैम ethnobotanical means प्लांट है उसका फ्यूचर उसका क्या क्या यूज कर सकते हैं. जैसे मेरा प्लांट है Justicia adatoda, तो इसका यूज हम जो लंग्स अपर रेस्पिरेटरी सिस्टम के जो डिजीज होती है उसको प्रिवेंट करने में इसका बहुत बड़ा योगदान है. तो इथनोबोटनी जो हम इसका यूज कर सकते हैं किस किस चीज में इसका यूज जैसे मेडिसिन और आपका.
I want only the meaning of the word ethnobotanical. This is your PhD research or MSc research? Hello.
PhD, ma'am.
PhD. Now you have to well know the meaning of the ethnobotanical. You have to tell so many properties of this plant that and so many alkaloids which are found in that particular plant. What is the use? What you have to say? Which alkaloids the human use for the drug making? Can you have any idea?
जी मैम.
कोई ऐसा ड्रग है जो इससे बनता हो जिससे कोई रिलीफ मिलता हो?
मैम क्यूनोलिन है जो मलेरिया जो फीवर
और कोई है कोई सिग्निफिकेंस और कहीं किसी जगह यूटिलाइज होता हो ये?
मैम मेनली तो जो अपर रेस्पिरेटरी सिस्टम के जो डिजीज कोज होती है.
अपर रेस्पिरेटरी सिस्टम.
जी.
What is basically lower respiratory system? Respiratory system तो एक ही होगा ना, उसको upper और lower में क्या define करना था? I cannot understand the meaning of upper and lower. Okay. The next one is the Kumud Gupta.
Yes, ma'am.
Share your screen, please.
Sure, ma'am. Is it visible? Hello?
Hello?
Yeah, am I visible? This screen is visible or not?
Your screen is visible, so you have to.
Ma'am, actually I have made a presentation also, and this particularly is my paper, so I want to read out through my paper. Is it fine, or shall I present only with the presentation?
ये word file है जी?
This is not a presentation. How you consider this word file in the presentation?
Okay, ma'am. I'll be presenting the PPT then. Fine.
Present paper.
Yes, I'll be presenting it. Sure. My particular topic is the role of science and tech in achieving sustainable development. And my name is Sumit Gupta, and I'm a research scholar at Desh Bhagat University. And basically, this is not related to my research area,
अच्छा,
but
आप इसका ना साइज थोड़ा सा इंक्रीज करो प्लीज डॉक्यूमेंट का।
सर कैसे मैंने स्क्रीन शेयर कर दिया वो स्लाइड शो करूं सर इसको?
ये पूरा पेपर है आपका, रिसर्च पेपर है ये?
नहीं सर, मैं दूसरी स्क्रीन प्रेजेंट कर रही हूँ। 1 मिनट, आई एम डूइंग इट अगेन। 1 सेकंड सर। Sir, is the PPT visible now? Hello?
हेलो, हाँ जी, अब ठीक है। हाँ जी,
प्रेजेंटेशन।
ओके ओके। तो इसको है ना, आप थोड़ा सा स्क्रीन को बड़ा कीजिए, F5। हाँ, अब ठीक है। ओके।
So basically, yeah. So my particular topic is role of science and tech in achieving sustainable development, and my name is Sumit Gupta, and I'm a research scholar at Desh Bhagat University currently. So, what do we understand by sustainable development, and how am I going to relate it with science and tech? Are two different things. So basically, we all know that sustainable development means that there are a lot of resources in the universe currently, and how we utilize those resources to the minimal is known as sustainable development. And how science and tech can be helpful in achieving that is what my presentation is all about. So I have done a lot of pictorial representation to make you understand the concept. So please bear with me. The very first slide says that what is sustainable. So this is a combination of two words, sustain and able, that is ability to sustain. So any resource, like for how long, what, and whatever resource it is, for how long we are going to use it is known as sustainable. And how the science and tech and sustainability are related is that, like, we all are human beings, and our actions on earth are going to affect the sustainability. So it's like a parasite and a host kind of a combination of technology and sustainability. So if we see, there are a lot of factors which depends upon health, happiness, and prosperity, and all the things which we want to use, or if we are going to use them in excess, will obviously lead to the decreasing of these three factors. So this is particularly a correlation in happiness, prosperity, and health. Like if we are going to exploit any particular resource, then we will obviously be not be leading to sustainability. There are a lot of policies which the government of India and of other countries is trying to apply to reduce the amount of the degradation of resources. And traditionally, the goal of public policy has been progress. Example, the 5-year plans are there, which our government has also established for a continuous movement to a better future and for material prosperity, that is greater goods and conveniences availability to humanity. So there is a particular goal which has been set by the government of India, and that includes the 5-year plans. This particularly is one of the examples to achieve sustainability through a particular flagship of programs. So if you see that through this policy, we have seen an unlimited growth and infinite growth rate, like by the application of these 5-year plans. So this is one, and the consequences are like growth requires resources, and it is fed to the machines and given out as products. So the 5-year plans, as one of the schemes, has led to a bit of reduction in those resources. And everything in nature has a limit on the rate of change of a particular variable. In engineering examples, rate of acceleration, loading, heating, pressure, etc., nothing can sustain anything with infinite rate. So beyond that rate of endurance, everything would obviously fail or react in an adverse reaction. So we have to apply certain policies, certain protocols to lead to the less advancement of these natural resources. So when nature reacts, if we are going to overexploit anything, then this is how the nature would react. We will be suffering from cyclones, from floods, which we are recently facing in Himachal Pradesh and other areas of Uttarakhand, and in a lot of other places of India and across the world. We will be facing, fire hazard, unprecedented growth in the weather, or the weather format would change. So this is what and how the nature would react if we would overexploit the resources. So the PPT basically represents the first thing, that is the 5-year plans have tried to reduce the overexploitation of resources, and the secondly, that how the nature would react if we would overexploit it. Then this is the graph which shows the global nature of disasters which have increased from 2010 and to current situation till 2009. So how do we react? It is our science and tech and our responsibility collectively to, to avoid the unprecedented challenges of the nature. So we can, control it by using the maximization of resources, and by continuing with the current model of development, entire nature and hence obviously the entire human race itself is threatened at the level of existence itself, and working towards a development model or more that focuses on holistic growth without resource exploitation and utilization rate would, account for replenishment. Means the potential of exploitation would reduce. As we can see, the sustainable development says that betterment of humanity in particular and ecosystem in general that balances the resource extraction is required, and waste generation rate with the replenishment potential of the earth. Like whatever waste we are generating should also be, controlled. So the glimpse of how the world has changed over the years by using a lot of science and tech for sustainability is in the next slide. We can see that since 1979, more than 20% of the polarized cap has melted away. And for this reduction, for this kind of melting, or for this kind of overexploitation of the earth's resources, we are trying to apply a lot of protocols. And one of them is the Stockholm Declaration, and which is a 1972 declaration, and which says that the power to transform this environment is in countless ways, is an unparalleled scale. Like we'll have to do certain measures to, reduce the environmental damage. The second is the Club of Rome. That is also a book which is published by the 1972 Stockholm model. This also says that we should be, minimally interacting with the nature's resources and not exploiting it to a lot of extent to, create a sustainable environment. Likewise, we have a lot of protocols and summits, like the Rio de Janeiro summit is there, the Earth Summit is there, and various conventions have been formed so as to, kind of reduce the overexploitation of resources in science and tech. Carbon trading should also be, it allows countries that have emission units to a particular level, and there should be clean development mechanism as stated by the Kyoto Protocol, and there should be a joint implementation of all these policies for a better utilization of science and tech. And this particular slide says is about the Millennium Summit, which happened in New York, and it states that how can we eradicate all these factors by sustainable development. So it is saying that over a period of 30 years from Stockholm to New York Millennium Summit, a fluctuation was seen in the degree of importance to be attached to economic growth, poverty, eradication, and environmental concern due to arrival on the on-scene developing and developed blocks. This, created a relook to the definition of sustainability. So this slide says that the limit of the growth of population or economy recognized due to resource dwindling, calling for a
असल में just conclude करूँगा.
Okay. So in short, this particular slide is the crux of the entire thing, that environment, sustainability, and social and economic factors are all correlated, and how science and tech has transformed it is a very big deal. Like for example, I'll give you a very recent example. Like wireless technologies are there, and a lot of smartphones we are using, and a lot of other techniques we have implemented in our current situation so as to reduce the exploitation or over exploitation of resources. So my entire paper and the entire presentation is about what are the policies, what are the protocols, and how we as human beings have used the resources to limit the over exploitation of the resources to create a sustainable happy development or a happy environment on earth. So this is the last slide. You can say that this depicts that it depends upon us, that we are human beings and the nature would react in adverse way if we would overexploit our resources. So that is all. A lot of factors are depicted in the slide, and a bit huge. So sustainability is all about giving up ownership and moving towards belongingness. The earth does not belong to us. We just belong to the earth for a short duration in the long span of life. Sustainability is about giving up wants in favor of needs. Thank you.
ओके ओके आप बताओ आपके जो रिसर्च इसका क्या सिग्निफिकेंस है?
सर बेसिकली जो मेरा पेपर है रिसर्च एरिया उसमें मैंने थ्योरेटिकल रिसर्च बेसिकली उसमें मैंने बहुत सारे रिसोर्सेज के बारे में डिपिक्ट करा है कि फॉर एग्जांपल वायरलेस जो सेंसर्स हम यूज़ कर रहे हैं आजकल वो जो स्मार्टफोन्स हम यूज़ कर रहे हैं उन सब की वजह से भी जो पहले अंडरग्राउंड पाइपलाइंस हमें बिछाने पड़ते थे या ऑप्टिक फाइबर यूज़ करने पड़ते थे वो कम हो गए हैं वी आर यूजिंग स्मार्ट लाइन्स एंड स्मार्टफोन्स दिस इज ऑल्सो वन ऑफ द एग्जाम्पल्स हम सस्टेनेबिलिटी के जा रहे हैं लाइक वी आर नॉट एक्सप्लॉइटिंग द अर्थ लाइक हमें डिगिंग वगैरह नहीं करनी पड़ेगी या फिर हमें ऑप्टिक फाइबर्स का ज़्यादा यूज़ नहीं करना पड़ेगा जो जिसमें बहुत मटीरियल लगता है तो स्मार्टफोन टेक्नोलॉजी इज वन ऑफ द एग्जाम्पल्स ऑफ साइंस एंड टेक अलॉन्ग विद सस्टेनेबिलिटी लाइक वो कार्बन इम्प्रिंट हम लोग को अपना कम करना चाहिए जितने भी हम रेफ्रिजरेटर्स यूज़ कर रहे हैं देर ऑल हैविंग दिस CFC लिमिट मतलब उसमें कार्बन एमिशन के बारे में एक रेट होता है उसके बाद ही यू कैन यूज इट सो basically it is a theoretical research like I have collected a lot of data from here and there and protocols के बारे में study करा है कि हम sustainability को कैसे implement कर पा रहे हैं through science and tech. So this is basically what my research area is in this particular paper.
ओके आपकी कोई पब्लिकेशन आई है?
सर इसको अभी मैं इसे currently I'm pursuing PhD and it is I have done other publication and one of them is in process currently.
ओके ओके बस ठीक है आप अपनी स्क्रीन को बंद कर दो.
थैंक यू सर.
ओके
because of the ability of these materials to distribute the cations over these two sites A and B, the properties of the fluids can be tuned. So this is the basic model Niels two sublattice model that how we can calculate the magnetization in the case of right materials. So we have some spin in the up direction, some spin in the down direction, then the net magnetization will be magnetization at the B side minus or the difference between the magnetization at B and A side respectively. So this is how the Needle to sublattice model can be used in the case of hexagonal structure, where we have different kind of the sites there, tetrahedral as well as octahedral. In addition to this, we have trigonal bipyramidal structure also. So this is the formula. Conclude. ठीक है. So, okay, at the end, I would just like to say that how these materials can be. Uh, so, this is the role of magnetocrystalline anisotropy, which is our paper, and we have studied the cobalt substituted Friday in this case, and we have seen that with the increase in the cobalt, the magnetic anisotropy is increasing. So, this paper was published in 2013, and this was given up by us because we have got the increase in the saturation magnetization while substituting the cobalt in place of the ferric ions, in spite of the fact that these have lower magnetic moment as compared to the Fe3 plus ions. So recently we have published another paper where we have gone for the ZFCFC curve, and we have shown that with the increase in the cobalt, we are having increase in the blocking temperature, which is responsible for the anisotropy, and ultimately this is responsible to increase the magnetization of the material. However, because another talk is also on the line, so we have published few books. These are our books which are available in the market. So 3 books we have edited, and rest of the things can be taken from there. So these are my research collaborators. So thank you very much.
for metal layer battery and gel polymer electrolytes for metal layer battery. He has experience teaching and research. He contributed in various international and national journals and has published books. So, sir, I present you presentation. Dr. I start.
screen, right? Okay. So my name is Rajneesh Thiman. I'm from Malvi National Institute of Technology, Jaipur. So I will talk about nanomaterials for energy conversion and storage. Previously, I thought to talk about gel polymer electrolytes, but since the focus of this workshop conference is on nanomaterials, so I slightly changed my topic. So here you can see the slide. It shows the significance of energy in the whole world. For example, the total energy consumption of the whole world is around 14 terawatt at the moment, or by 2010, it is supposed to triple by 2015. And one can see the different countries. For example, US consumes the most of the world energy, and then China comes the second, while India is also at the fourth number. So most of the energy is being produced from different sources such as nuclear, natural gas, etc. Oil contributes to 34% of the world consumption of energy or production of energy. And most of the oil is being used for transportation. Here you can see 28% of the total energy is used for transportation, and more than 80% of the oil is being used for transportation purposes. And it also contributes to 40% of the total CO2 emission. And there are several other reasons where the fossil fuels which are being used for transportation that causes damage, for example, to the environment. Moreover, there are limited resources, and global instabilities can also create chaos in the whole market. So one has to look for alternatives. Biofuels could be another alternatives, but they do have some sort of side effects, such as food insecurity. Some technical issues are there. They are less efficient. Moreover, the destabilization of ecosystem could also be caused by biofuels. So we have to look for other alternatives as well. So electrochemical energy conversion and storage, that's one of the most sought sources of energy, especially for transportation. So if I say electrochemical energy conversion, then I'm referring to fuel cells. When I'm talking about electrochemical energy storage, then it means we are talking about batteries and supercapacitors. Am I audible? Am I audible?
Yes, sir, you are audible. Please go ahead.
Okay. I cannot hear anything, so that's a bit confusing. Anyway, so I'm continuing. So here this slide shows the significance of nano in case of energy storage. For example, here we have the solar cells. So where the whole research is being focused towards decreasing the size of the materials, and one can have better efficiency by using the nanomaterials. Here, this slide is about the battery systems. I'm going to talk about, or I will show some of the results of battery research and fuel cells which we are doing here. So when I talk about battery systems, and their utilization is mostly in the electric transportation, solar energy storage, home inverters, communication systems, etc. If we talk about the efficiency in case of batteries, then lithium air, for example, is one of the kind of battery which have the highest theoretical specific energy density. The lithium ion batteries are also there, but they have very less energy density, theoretical energy density compared to the lithium air. Moreover, if we calculate the practical energy density or practical specific energy density, then still one can see that zinc air batteries, lithium air batteries, or we could also say the metal air batteries, they are speculated to have high energy density compared to lithium ion. So this is the reason that I'm working on metal air batteries. These are the kind of batteries where the cathode consists of the air. Basically, it's a porous material where the air, oxygen from air, that gets reduced, while the anode material that gets oxidized, and the ionic transportation goes through the electrolyte, while the electrons, they goes through the outer circuit. One can see it here. So this way we can, when the battery is being discharged, electrons flow from anode to cathode, and we can use it for our applications. So the depending upon the different metal anodes, for example, lithium, zinc, magnesium, sodium, one can have the lithium air battery or zinc air battery, magnesium air battery, and so on. So here is the mechanism where oxygen goes to the air cathode, and it gets reduced to produce the hydroxy lines, where hydroxy lines, they transport through the electrolyte, which could be liquid electrolyte or some sort of quasi solid electrolytes, for example the gel polymer electrolytes, and they react with the metal over here to produce the metal oxide. So when the battery is being charged, the whole of the electrochemical reaction that is forced to go in the opposite direction. So why metal air batteries? I already told about the theoretical energy, specific energy density, very flat discharge voltage. This is very important, because if we have to use a battery, then it should be operated at a constant voltage. So we need to have a flat discharge voltage all the time. So they are speculated to have low cost. So one can figure out that these are not into commercial at the moment, but these are into research. So this is something about zinc air battery, and here we are focusing on different materials, different electrode materials, and we are using manganese dioxide, cobalt oxide, or silver RGO based nanomaterials for the electrode, where these materials, they act as the cathode. So these are some other different combinations which we are using for the air cathode. So this is something about the battery device which we fabricate in our lab for our research work. So in case of these batteries, the cathode material is basically coated over a nickel foil. One can see the nickel foil over here. So nickel foil act as a current collector, where the material is coated over it. So this is the complete cathode, and we use we use zinc metal as anode, and we have this zinc foil for the anode. Here you can see this is the quasi-solid or the gel polymer electrolyte which we use in this battery in our lab. So the gel polymer electrolyte, this is also important, and one can see the polymer, it have pores inside. So the KOH is the most commonly used 6 molar. For example, the 6 molar KOH is the most commonly used electrolyte. So basically the electrolyte 6 molar KOH, that is being soaked, or the polymer is being soaked with the 6 molar KOH to make it ready for the battery operation. So this is the scanning electron microscope image of our polymer. As manganese dioxide is one of the electrode material which we are using, so manganese dioxide, we grow manganese dioxide nanowires. So one can have this web of nanowires that look like a kind of fabric. And here the size of these nanowires, that is the average size, we could say, that is around 30 to 35 nanometers. This XRD shows that we have a we confirm the synthesis of manganese dioxide material. Am I still audible? Because I cannot hear anything. Am I still audible?
Yes,
Okay, so this is something about the gel polymer electrolyte, because we need a prepared electrolyte for the battery operation, and we used several combination of polymers, for example, polyvinyl alcohol, carboxymethyl cellulose in different ratios and then different concentration for growth. And then we optimize the thickness of the polymer as well, gel polymer electrolyte, and we can figure out that if we use the 4 thickness of the electrolyte, then we get the most number of cycles. And the cycling is also directly proportional to the 6 molar KOH, which is soaked inside the gel polymer electrolyte, and we could see that the maximum the amount of KOH uptake is there, then the number of cycles are also the maximum. And similarly, when we have the maximum uptake of KOH, we get the minimum or the least charge transfer resistance, which is desired for this battery operation. So this way we have optimized it, and one can see we have also optimized the thickness. So when this gel polymer electrolyte is ready, we run our zinc air batteries, and we also calculated the specific energy density, and we get a specific energy density of around 700 milliampere hour per gram of the zinc which is being consumed. We also look at the degradation of the gel polymer electrolyte, and one can see um, the damaged or degraded polymers. However, they are still good enough for several number of cycles. For the time being, we are getting around 300 cycles at the moment. So this is just a demonstration of these batteries. So another material which we are looking is the composite made of manganese dioxide, RgO. And this combination, the particle size or the crystallite size which we get, this is actually corresponding to the manganese dioxide. The size is around 23.6 nanometers. We use different combinations. For example, here in these two pictures, one can see from this pointer. Here, whatever the nanowires which we get are usually mostly of the uniform size of around 30 to 35 nanometers, while if we increase the amount of manganese dioxide, for example, in this picture, the manganese dioxide used is around 30%, while 70% is reduced graphene oxide. While in this picture we can see a different morphology compared to this one, compared to the previous one, and in this case the manganese dioxide used is around 50%, while RgO is also 50%. One can see that the manganese dioxide nanowires, the length of these nanowires that get decreased, and one can see there are some of the nanowires which have around 5 micrometer length, and the size or the diameter which was originally in the beginning was 35, that gets also some of the nanowires or nanoviscors which we get, they have large diameter compared to the other ones. And again, when we increase the amount of manganese dioxide bit more, then we even see the bigger wires. Then one can say that these are micro wires instead of nano, because some of the wires, they do have a size or diameter of around 2 micrometer. However, the original web of nanowires that still is there, but one can well, some of the wires, they also get increased in the diameter. So one can see the different morphologies. We also characterized the nanowires with transmission electron microscopy, and one can see the TEM images. So here again, the scale is around 100 nanometer, and this confirms the size of around 35 nanometer diameter. Similarly, this is again at different location, actually different composition. So this one is 50% MnO2 and 50% RgO, and one can see a slight increase in the diameter. So we have the high resolution image as well, and one can see these individual planes of the crystalline manganese dioxide. And for example, in this picture we can see the interlayer distance between these the crystalline planes is around 7.4 angstrom. And in the lower one, this is 3.6 angstrom between the two adjacent layers. And these two, for example, this one is around the 7.4, while this one is 3.6. So this means they both of these nanowires, for example, or the location of nanowires, they correspond to different reflections or different planes in the crystal. So here are some of the discharge charge curve. So one can see the this is the zoom in of the first or second cycle, and one can see the battery in the beginning is charged and it have a potential of around 1.4 volt, and when we discharge, the plateau goes up to 1.3, which slightly keep on reducing with the time. And here is the first discharge, well, actually second discharge of this battery. And here one can see this one, this is the second charging of the battery. So when we have charged the battery, the electrochemical reaction is forced to go the other way as that of the discharge. So this way it keep on going. And when we look at the whole graph, then for example in this battery we have discharged it up to 50 hours, which consists of around 153 cycles. So here we have a discontinuity in the discharge profile due to some of the reason which I may not discuss here at the moment. So this is the last discharge. By the way, we are actually discharging this battery until 1 volt only. So until 1 volt, we are getting 153 cycles. So it means that when we are discharging it up to 1 volt, then it means the device which it can run, which this battery can run, should be operatable at 1 volt. Now we have different combinations, and one can see the discharge, the charging curve. It's all continuous and it goes up to 144 cycles. And again, the initial discharge plateau is around 1.3 volt. So similarly, some other modifications, 50-50 composition of MnO2 and RGO, we could go up to more than 50 hours, which makes 150 cycles. So again, the first cycle and the last cycle. Then again, the different composition, MnO2 is 70% and 30% is RGO. Then we can go up to 180 cycles when discharged up to 1 volt. If we are discharging this battery until much lower voltage, it can go to higher number of cycles. But the thing is that batteries should be operatable at operational. They should be operational for a particular device at a particular voltage. So most of the devices, they are operational around 1 volt or slightly higher than that. For example, if we have a battery which is being used in two-wheelers, that is operational at 12 volts. So which means that if we have to use this battery, then we need 12 zinc air batteries in series. So this is something about the nanomaterials. I'm only talking about the nanomaterials which we are using in the batteries. By the way, just to tell a little that when we are using this manganese dioxide basically acts as a catalyst to catalyze the oxygen reduction reaction which takes place at the cathode. I'm still audible? Can you please confirm?
Yes, sir.
Okay, so now I will move to the section of fuel cells. So basically, I'm working on polymer electrolyte membrane fuel cells. So in case of fuel cell, hydrogen is used as a fuel, and the oxygen reduction reaction takes place at the maybe I have this battery. Yes, I have this schematic. So this, for example, this fuel cell, here in this case, we use hydrogen as a fuel, which oxidize at the anode, and the electron is transported through the outer circuit and the load or the particular device where it's being used, that can be put inside this in this circuit. And the protons, which are the ionic charge carriers inside the electrolyte, they get transported through a proton exchange membrane. So most commonly used membrane, especially in commercial fuel cells, is the naphthalene. So proton, when it is transported through, it goes at the cathode, and it reacts with the oxygen which is available from the atmosphere. It reacts with oxygen, or basically the oxygen reduction reaction, to produce water. In the process, the electrons which are coming from outer circuit, that they are being used in this reaction. So here we can see the design of a polymer electrolyte membrane fuel cell. It look like this. So we have these steel plates, one at one side and the other one on the other side. They are basically cathodes and anodes. So this is actually the schematic design, how it look like. So here we have this porous cathode, and this is the porous anode where oxygen is being fed. And actually, I'm not talking about some of the manufacturing or some of the actually there are several things which are used inside. For example, the membrane electrode assembly, gas diffusion layer. So they are several different things which are all inside this. So one can also see this schematic. So these are the steel plates which you saw in this picture, and we have the current collector. We have different gaskets, etc. We have these gaskets because when a fuel cell is operational, it produces water as a byproduct. So water is there, and moreover, we need to humidify the polymer membrane which is inside. So water management is also a very important thing. So it means the whole of this fuel cell that need to be leak through as well. So one need lot of gaskets. So this is one of the gas diffusion layer, which is gas diffusion layer is actually used on both sides, on the cathode as well as the anode. So ultimately, this is the only electrolyte, and rest of the stuff is the arrangement for all the cathode, and here it's for the anode. One can see the water which is coming out. So and water which is coming out. So that's also an important thing. However, I'm not discussing about that. I will discuss about the cathode material. Cathodes are my main interest. So platinum on carbon, or basically platinum, 40% of platinum on carbon black is the commercially used electrode material for the fuel cell, and platinum is actually the catalyst and the most efficient catalyst as of now. The other catalysts are palladium, ruthenium, etc., but they are not that efficient. But as you can see, the platinum is used as a catalyst. So one can see that the cost of the commercial fuel cell, or the cost that rise, and it's around the 40% of the cost that is caused by the cathode and anode, which use platinum as catalyst. And as I said, the 40% of platinum on carbon black. So 40% of platinum is too high. So it means if we have to commercialize at a large scale. By the way, fuel cells are already in commercial use. So you might have heard about the fuel cell based electric vehicles. Earlier, I think Tata also launched a bus based on fuel cells, but it's the cost which is high. And another problem with the fuel cell is the degradation of the electrode material. Carbon is the most commonly used because it's the most convenient. It's a good electronic conductor, and moreover, its cost is low. So that's why it's the most usable as of now. But since it suffers from some drawbacks, for example, it corrodes especially in the especially in the harsh electrochemical environment when the fuel cell is working. Okay, so we are focusing on alternative materials. So we are looking at silver on reduced graphene oxide as one of the electrode material. But one can assume that we are using silver on RGO. So this material is not as efficient as platinum on carbon black. But the important aspect of this is to reduce the cost, of the cathode and anode materials, which will ultimately help to commercialize it on large scale. So here is one of our materials. So one can see these are the transmission electron microscope images. I think I can show you some scanning electron microscope images. So here one can see this is the reduced graphene oxide, and here we have these different graphitic planes or graphene sheets, one can say. And whatever the white material you see on this scanning electron microscope images is actually the silver. So there are different magnifications. This one, the scale in this case is 500 nanometers, and one can see the silver nanomaterials over this. And this is the transmission electron microscope image. I will go back again. So here in this image, you can see this dark spherical particle, and the scale is 5 nanometer, which means that the diameter of this nanoparticle could be around 25 nanometers. So one can see from this image that the silver particles are uniformly distributed over it. So it takes time or times to optimize the different parameters to have a uniform distribution of these particles. So that's the other story. So here these are the high resolution images, and in this case one can see uh, the different planes, different planes, and these planes are basically the different graphene sheets. And the distance between the graphitic sheets are 0.34 nanometer, which means 3.4 angstrom. So this is one of the silver particle. And here you see the parallel lines. These are actually some sort of defects which we call uh, twins in this case. Anyways, so these are the different compositions. So in this case we have silver 25% and 75% of reduced graphene oxide. In this sample, we have 40% silver and 60% of reduced graphene oxide, and this is one of these are the four transmission electron microscope images. These are some of the parameters which we can calculate from here. Okay, so these are the scanning electron microscope images showing a relatively uniform distribution. These are the EDX images, where EDX imaging we have done, and here you can see this is the scanning electron microscope image which we use to map the different elements. So we mapped carbon, nitrogen, oxygen, and silver. So basically in this case the reduced graphene oxide, this was nitrogen doped. So one can see a relatively uniform distribution of nitrogen. So one can see these spots, so relatively they are quite uniform. And since the base material is reduced graphene oxide, so that's a carbon, you can see it's all uniformly distributed over everywhere. So oxygen functionalities, they are always there, so these are again quite uniform. By the way, the scale is around 8 micrometers, so the whole length would be around 35 nanometers. So here the main focus is on silver. So here the uniform spots, these shows that the distribution of silver is quite uniform. Similarly, the different composition and some of the oxygen reduction reaction results which we use to evaluate the electrochemical performance. Am I audible?
Yes, sir. You may continue, please.
Okay. Okay, so I'm continuing now. So here in this image, we have the linear sweep voltammetry. These are the linear sweeps. So basically, half of the half cycle of the cyclic voltammetry, one can simply use the linear sweep, which means only one cycle. So here these are the different compositions. So in this case, it's just the base material reduced graphene oxide, and here in this case, the silver is 10%. Here the silver is 25%. In these three, and here we have the other sample with 40% silver, and this one is actually 55%. Well, this is 55%. Yeah, 55%. So this is basically the TEFL plot. Actually, this is the linear sweep voltammeter, and these are the TEFL plots. So these are used to evaluate the electrochemical properties. So the earlier, these are the physicochemical characterization, and the main application part is for fuel cells. So we need to evaluate the electrochemical performance. So here one can see the limiting current on the z-axis. So the maximum, by the way, in case of linear sweep voltammetry, we use rotating disc electrode. So basically, this is a 3 electrode system where the cathode material or the working electrode is actually rotated at different rotational speeds. For example, we rotate it at 400 rotations per minute, 800, 1200, and up to 2000 RPM. So the oxygen reduction reaction is actually quite sluggish, and in order to evaluate the material, we need to remove the byproducts by the earliest. So that's why we use to rotate the cathode material. So one can see that higher the rotational speed, higher is the current and the y-axis. So so we use this current density. So we have used the surface area of the electrode to calculate the current density. So basically, limiting current density is there. I think I shouldn't go further about the limiting current, etc. But just to say that higher the limiting current current density, higher will be the electrochemical activity. So we compare it with silver 10%, and one can see the y-axis. It's up to 8 milliampere per centimeter square. When we use simply the graphene, this is only around 3.5 milliampere per centimeter square, and further we increase the composition, we get the increased limiting current, and these are the TEFL plots, which are again used to calculate other parameters. And let me summarize some of the parameters. Here, in case of RGO, the limiting current which we get is around minus 3.4 milliampere per centimeter square, while in case of silver 40%, we get a limiting current of minus 8.6. So higher limiting current is desirable. So that's why the one of the parameter, that is limiting current density, it concludes that 40% silver is 40% silver on RGO is the better material for this one. And here one can see the variation 3.4, higher content of silver 6.1, 6.4, and then the maximum is obtained at 8.6. Then it further start reducing when we increase the amount of silver up to 55%. Okay, so number of electrons is another parameter which we evaluate from these TEFL slopes, and I haven't taken enough of the introduction by considering the limited time for the presentation. So let me just say that the maximum number of electrons which are transported in the oxygen reduction reaction is actually 4. However, there are two different pathways for the oxygen reduction reaction, which are 2-electron pathway and 4-electron pathway. 4-electron pathway is the most desirable one. The 2-electron pathway is actually, in the case of 2-electron pathway, hydrogen peroxide is one of the let me say, the middle product which is being formed, and hydrogen peroxide is actually deteriorating to the electrolyte membrane. So that's why we avoid the we avoid the um we avoid the production of hydrogen peroxide, which means we try to get the materials with the higher number of electrons transferred. For example, in Ag55 we have 3.8 number of electrons. Similarly, in Ag40, 3.8 again, and for simply for RGO, it's 2.4. Well, you can see that 3.8 is actually a fractional number, which, I mean, electron cannot be fractional, right? But it's the evaluation where we calculate it for several products, which means there are several chemical reactions which are taking place. So some of the reactions are following the 2 electron pathway, and some of them are following the 4 electron pathway. So the average comes out to be 2.4. That's what it means. So smaller the number of electron transferred, higher will be the amount of peroxide produced. So it means in case of this as well, the limiting current is high in case of 40%, and relatively smaller amount of hydrogen peroxide is produced. Okay, so the onset potential is another parameter inside, and onset potential should be as low as possible. So the lower value of onset potential is desirable. Here, in case of Ag40, we are getting 0.85 volt, while in case of RGO, it is around 0.79. So here it's a little bit disadvantageous for onset potential. However, we are working on reducing the size of silver nanoparticles. The smaller the size of the catalyst material, for example, silver here, the higher electrochemical activity it is going to have. The simple the simple idea is just to have a high surface area available for the catalytic reaction to take place. So so this way we are working on it to reduce it further. We have got some success at the moment. And another thing is that we also calculate the electrochemical surface area, which is another parameter. So apart from these 3 parameters, we do use the electrochemical surface area. So for example, here the calculated values for silver with 40% amount is actually 25.2 meter square per gram. So here it means that 25.2 meter square of the area of the catalyst per unit gram as also. So it means 1 gram of catalyst material, the 25 meter square of this area is being used for the electrochemical process. So similarly, in case of Ag 10%, the specific surface area, electrochemical specific surface area is 11.1 meter square per gram. So it means a relatively less area in Ag10 is being used. So one need to have high value of all these parameters to have the higher or better electrochemical activity. So we use the cyclic voltammetry to calculate the electrochemical active surface area. For example, the oxygen reduction reaction for the that is used to calculate the area. Okay, so we use electrochemical impedance spectroscopy this is another parameter, electrochemical impedance spectroscopy. So we use this EIS to calculate the impedance. And impedance have different contributions. For example, one of it is series resistance, while the another one is charge transfer resistance, Warburg resistance, etc. So series resistance, or one could also say it's a solution resistance. So basically the combined resistance, by the way, the desirable resistance is the minimum. So here the values for this series resistance, this includes um the resistance, electronic resistance of the electrode material, the ionic resistance in the electrolyte electrode interface, which is formed over there. So less is the amount of series resistance, better it is. So here in this case, one can see uh, more or less the resistance is, the series resistance is more or less equal. While the charge transfer resistance, this is actually the resistance which occurs in the Faradic reaction. So Faradic reaction is the actual reaction which takes place and which controls the different parameters. So this is also desired to have as little as possible charge transfer resistance. So here we can see the silver with the content of 55% have the least resistance. However, the silver with 40% have the high limiting current, which is the most desirable in this case. So now another thing is that you would find, or whenever the surface area of the materials are used, the rule of thumb is that the amount of the nanomaterial should be in such a way that uh, the optimized area is as high as possible. So here if you see the 10% of silver, in that case the surface area of the silver nanoparticles is only 10%, so that is relatively less than that of the other ones. So here silver with 55%, you have, well, I showed you the previous slides which says that the higher the content of silver, higher is the particle size, which means that significant amount of silver, since that is inside the bulk, that is not being used, so it's going useless. So that is why you would always see a middle percentage that is the most optimized, and that's what we see here as well. So these are the Nyquist plots. By using them, we have calculated these parameters. Okay, so I think I should stop here. So that's all, and these are my PhD students and MSc students. These are so basically this is all of the results are obtained by these students. So thank you very much.
Thank you, sir, for such an informative talk. We appreciate your contribution in technology for attaining sustainability. And we thanks to you for sparing so valuable time. Thank you, sir. Thank you so much.
You're welcome, ma'am. You're welcome. So any questions or comments, discussion? If you have time, I'm available. We can discuss. If it's time shortage, still it's okay. We can, yeah.
Any question, audience? If any question is there from anybody, you can ask to the sir. Okay, sir. Thank you so much.
You're welcome, ma'am.
Okay, sir.
With this presentation, we have come to end of the day 2 of the 3-day national conference on empowering sustainability, bridging science, technology, and climate resilience 2024. You cannot go back and change the beginning, but you can start where you are and change the ending. Respected delegates and all the participants of the conference, on behalf of entire organizing committee, I convey a wholehearted thanks to you all for gracing and making the day successful. Thank you all. Thank you everyone.
We will meet tomorrow on the second day for the continuation of the conference. Thanks once again.